Thailand kemarin ini orang Amerika Perancis yang nah suruh kini merangkum ibu nih ibu Asma apa materinya dulu kalau Inggris itu dulu
Faculty of Mathematics and Natural Sciences, Universitas Negeri Makassar. The Faculty of Mathematics and Natural Sciences, Universitas Negeri Makassar has vision to excel nationally and globally competitive in developing mathematics and natural sciences and learning, as well as to produce competent graduates in their field, literate in information and technology, and entrepreneurial-minded in 2025.
Selamat datang kami ucapkan kepada Rektor Universitas Negeri Makassar, dalam hal ini diwakili oleh Wakil Rektor Bidang Akademik Profesor Dr. Hasnawi Haris MHum.
Sini sumpul lolo pada tudam majejereng kurusumanga pada ipatarakan niat kata gau marajana biologi. Nah pada tuang kana tulang marengkali ngari tahu wacana lino ajaga jaga makanada tolino baratudak kimareng kalinga. Jaga kedo salama tuang kana patarakai pamasena puang Allah dewata.
first Chemistry International Conference and the second International Conference of Life Science and Biology Education. So that was a traditional dance for special guest in South Sulawesi culture. The Honorable Rector of Universitas Negeri Makassar, represented by Vice Rector for Academic Affairs, Professor Dr. Hasnawi Haris MHum. The Honorable Dean of the Faculty of Mathematics and Natural Sciences, Universitas Negeri Makassar, Associate Professor Suwardi Anas, MSc PhD. The Honorable all keynote speakers and invited speakers, the Honorable our distinguished guests, and the Honorable all participants joining our event today. Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. First of all, let's raise our praise and gratitude to the Almighty God who still gives us mercy, blessing, and health, so that we are able to attend the first Chemistry International Conference and the second International Conference of Life Science and Biology Education. Ladies and gentlemen, let's start our event today by reciting Basmalah. Bismillahirrahmanirrahim. The next agenda, singing the national anthem of the Republic of Indonesia, Indonesia Raya. We would like to invite all participants joining in this room to stand up, and those who join this event virtually, please pay attention. Please start. Thank you. And for the next event, I would like to invite the head of committee to deliver a greeting for Dr. Rihardlo. The time is yours.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Honorable Rector of University Negeri Makassar, represented by Vice Rector for Academic Affairs, Professor Dr. Hasnawi Haris MHum. the Honorable Dean of the Faculty of Mathematics and Natural Science, UNM, Dr. Andes Suwardi Anas, MSc, PhD. The Honorable all of the Vice Deans of the Faculty of Mathematics and Natural Science, Makassar State University. The Honorable, our keynote speakers. First, Professor Nathalie Bourgonon, University de Bretagne, Sud, France. Second, Prof. Kazuyuki Inubushi, Tokyo University of Agriculture, Japan. Third, Dr. Hee Eun Chu, Macquarie University, Australia. Fourth, Professor Christopher Thompson, Australia, Monash University. The Honorable Invite Speaker, Professor Sonia N. Martin, Seoul National University, South Korea. Professor Dr. Fahrul Zaman bin Huyop, University Technology, Malaysia. Assistant Professor Sirinapa Kichkwakul, Naresuan University, Thailand. Profesor Oslan Jumadi, PhD, Universitas Negeri Makassar. The Honorable all of lecturers of Universitas Negeri Makassar. The Honorable all participants, face-to-face and online, joining in this international conference. First of all, let's praise our praise and gratitude to the Almighty God, who still give us mercy and blessing, so that the second International Conference of Life Science and Biological Education, organized by Biology Department, and the first Semestry International Conference, organized by Semestry Department, Universitas Negeri Makassar, can be successfully held today. We also send salawat and salam to our beloved Nabi Muhammad sallallahu alaihi wasallam. May Allah subhanahu wa ta'ala blessing him with his family, his friend, and his followers. As the head of committee, I would like to deliver my sincere appreciation to all keynote speakers and all invite speakers for their willingness to spend their time with us at the second International Conference of Life Science and Bioeducation and the first semester International Conference to share knowledge and insights of the field of biology and chemistry. There are a total of 370 participants joining this conference, both offline and online participants, including 105 parallel presenters. Presenters joining this conference are researchers, teachers, educator, and students from various institutions, including from Hiroshima University, Japan, Monash University, Australia, and several universities across Indonesia, such as Universitas Gadjah Mada, Universitas Pajajaran, Universitas Hasanuddin, Universitas Negeri Malang, Universitas Negeri Makassar, Universitas Islam Makassar, Universitas Bosowa, Universitas Sulawesi Barat, Universitas Halu Oleo, UIN Wali Songo Semarang, Universitas Riau, Universitas Negeri Medan, Universitas Tanjung Pura, Universitas 19 November Kolaka, Universitas Nusa Nipa, Universitas Borneo Tarakan, STMKG, College of Meteorology, Climatology, and Geophysics, Senior High School from South Sulawesi, and also from National Research Agency and Innovation, PRIN. For the chemistry conference, there are 350 participants. We do hope that knowledge sharing through this conference can bring positive impacts to improve the quality of research and education, particularly to foster collaboration among researchers in the field of biology and biology education and chemistry. On behalf of the community, I would like to apologize if there are weaknesses during this conference. Wassalamualaikum warahmatullahi wabarakatuh.
Ladies and gentlemen, for the next agenda is regards from the Dean of the Faculty of Mathematics and Natural Sciences, Universitas Negeri Makassar. Ladies and gentlemen, please welcome Associate Professor Suwardi Anas, MSc, PhD.
Mana konsep saya?
Oh, the greetings. Oh.
Oh, iya. Hampir saya tidak sambutan. Ya, baik. Ya, kaget juga. Ya. Baik, Bismillahirrahmanirrahim. Selamat pagi. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
First, the Honorable Rector of Universitas Negeri Makassar, today presented by Vice Rector for Academic Affairs, Profesor Dr. Hasnawi Haris Efung. Thank you very much, Prof. Bersedia datang untuk membuka acara kami. Ini penghargaan yang luar biasa, Prof. And then the Honorable, our keynote speakers for Chemistry International Seminar, Professor Barbara Oakley from Auckland University, USA, maybe join in the virtual Zoom. Yeah. And then the Honorable, our keynote speaker for International Conference on Life Science and Biological Education. The first is Professor Natalia Bergoman. Thank you very much. From University, the difficult to spell, Professor, because maybe French language. And then we have Professor Kazuki Inubushi from Tokyo University of Agriculture, Japan. Thank you. Ohayo gozaimasu, Inubushi sensei. Watashi mo Osaka Buritsu Daigaku Shishugo shimashita. Junin kami. Saya hanya mengatakan selamat pagi, Profesor Inubushi. Saya juga alumni dari Osaka Prefecture University Jepang, sekaligus memperkenalkan diri bahwa alumni dari sana ya 15 tahun yang lalu. Itu ya, itu yang saya bilang sama Profesor Inubushi. Ya, sudah doktor tua. Ya. And then we have Dr. Ewan Chu from Mercury University, Australia. And then the Honorable, the invited speaker for a Chemistry International Seminar. And today we have 6 speakers. The first is David Reed from Academy, Director of English Language Teaching Center, the University of Sheffield, United Kingdom. And then we have Professor Rio Honda, Vice Director of Environmental and Energy Technology International Course, Kanazawa University. Come here. Join will in the virtual ya. Ya, kebetulan 2006 saya di universitas ini konferensi juga, sangat cantik kotanya Kanazawa. Ya, salah satu kota tercantik di Jepang. And then Profesor Christopher Thompson, thank you very much. Ya, Profesor Christopher, dan beliau sudah makan pisang epe tadi malam. Ya, pisang epe, Prof. Ya, Profesor. Ya. Yeah, Director of Education of Biomedical Discovery Institute, Monash University, Australia. And then Dr. Ronnie Horrocks, Postdoctoral Fellow, Department of Chemical Engineering, University of Arkansas, UK. Luar biasa semua pembicaraannya. And then Dr. Jonathan Taylor, Department of Chemical and Environmental Engineering, University of Nottingham, UK. And then Profesor Dr. Muhammad Daniel, MSc. Wah, ini dosen kita. Ya, alhamdulillah. Dari I think Professor Daniel is the head of study program, chemical education, postgraduates of University of Makassar. And then the honorable, the invited speaker for ICOLITE. The first is Professor Sonia and Martin, Seoul University, Seoul National University, South Korea. And then Professor Dr. Fahrul Zaman di Hubei, University of Malaysia. Terima kasih. Prof. Fahrul telah bergabung. Ya, kami dengan Pak Musawir sudah 2 kali nginap di dalam kampus UTM. Ya, luar biasa kampusnya. And then Associate Professor Sri Napa Kiskuko, Naresuan University Thailand. Mungkin Pak Musawir yang kontaknya ini, ya, karena beliau banyak berinteraksi dengan teman-teman di berbagai universitas di Thailand. And then Profesor Osla Jumadi PhD. Ya, tadi banyak sekali fansnya. Ya, ya seperti itu. The Honorable Vice Dean, Head of Department, Head of Study Program, Head of Laboratory, and all participants, especially for our students joining today. Good morning, ladies and gentlemen. First, I would like to take this opportunity to express my sincere appreciation to all keynote speaker and invited speaker for accepting our invitation to share the recent finding and best practices here. At the first Chemistry International Conference, CIC, organized by Chemistry Department at MIPA UNM, and then the second International Conference on Life Science and Biological Education organized by Biology Department of MIPA UNM. Ini luar biasa ya. 2 International Conference Power 1 kami melaporkan di hari yang bersamaan. Karena di MIPA ini semua departemen prodi wajib melakukan conference. Kalau tidak bisa internasional, nasional pro dalam setahun. Supaya atmosfer kehidupan ilmiah dalam kampus itu semakin berkembang. And then I would like to take this opportunity to thank the chair of the first CIC, Sulfikar PhD. She also graduated from Japan, and Associate Professor Wiharto. Ya, terima kasih Pak atas terlaksananya program ini dengan baik. This international seminar are not only expected to be a forum to disseminate the latest research finding in the field of chemistry, biology, and education, but it is also expected to be a networking forum to foster more collaboration among researchers, academics, school teachers, and also graduates, students joining this event. Behind the implementation of this international conference, this is household commitment and outstanding cooperation of the executive committee. Therefore, to the whole team of the executive committee, I really thank you for hard work and enthusiasm for the success of the first CIC and the second of ICOLITE. I would also like to take this opportunity to thank for all participants, and finally, as the Dean of the Faculty of Mathematics and Natural Science, University of Negeri Makassar, I want to congratulate all participants joining today. And I am very sorry, especially for our special guests from abroad countries and all participants, because I cannot full time here. And maybe Pak Wirsatu juga because in the same time we have accreditation assessment from the Ministry of Education, Culture, Research and Technology of Indonesia at the same times. Yeah. And I think enough our speaks today, and please enjoy your time during the conference session. And I hope Pak Wirsatu untuk membuka secara resmi memberikan sambutan dalam konferensi ini. Billahi taufiq wal hidayah. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much for the Dean of the Faculty of Mathematics and Natural Sciences, Universitas Negeri Makassar. Ladies and gentlemen, it is our honor to have the Rector of Universitas Negeri Makassar, represented by Vice Rector for Academic Affairs, to deliver a welcoming speech and also to officially open the first CIC and the second ICO route. Please welcome Professor Dr. Hasnawi Haris MHo.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Saya pikir tadi ini intermezzo saja. Saya pikir tadi Pak Ketua Panitia itu dari Jepang juga. Rupanya saya periksa banget, ternyata Pak Wiharto. Iya. Pak Dekan tadi bisik, kayaknya dari Jepang juga ini Pak komite chairman-nya. Baik, first of all, praise to be to Allah Subhanahu wa Ta'ala for His blessing and peace be upon Prophet Muhammad Sallallahu Alaihi Wasallam as the messenger of Allah. This time, allow me to apologize on behalf of the rector of UNM because he needs to others agenda, and this time so he asked me to represent him. The Honorable Vice Rector, Director of Postgraduate Programs, Heads of Institute, Deans, wabil khusus dengan Matematika dan Ilmu Pengetahuan Alam and Head Study Programs at UNM. I would like to welcome the keynote speakers and invited speaker for the second international conference on life, science and biology education. The keynote speakers are Professor Nathalie Bourgnon from Southern Brittany University, France, Professor Kazuyuki Inubisi from Tokyo University of Agriculture, Japan, and Dr. Hai Yun Chu from Macquarie University, Australia. And the invited speakers are Professor Sonia N. Martin from Seoul National University, South Korea, Profesor Dr. Fahrul Samam bin Hoyuk dari University Teknologi Malaysia, Assistant Professor Srinapa Kichkuakul from Nation University Thailand, and Professor Oslan Jumadi PhD from Universitas Negeri Makassar. I also would like to welcome the keynote speaker and the invited speakers, the first Chemistry International Conference. The keynote speaker is Professor Barbara Oakley, Distinguished Professor of Engineering, Auckland University in Rochester, Michigan, UK. And the invited speakers are Professor Christopher Thompson from Director of Education at Biomedicine Discover Institute, Monash University Australia, yang tadi sudah disampaikan Pak Dekan, sudah mencicipi pisang epe tadi malam. Thank you. Professor David Reed, the Academic Director of English Language Teaching Center, the University of Sheffield, UK. Professor Rio Hondo, Vice Director of Environmental and Energy Technology International Course, Kasanawa University, Japan. Dr. Ronnie Horax, Postdoc Fellow at the Department of Chemical Engineering, University of Arkansas, UK. Dr. Jonathan Tew, from Department on Chemical and Environmental Engineering, University of Nottingham, UK, and Professor Dr. Muhammad Daniel MSc dari Universitas Negeri Makassar. Ladies and gentlemen, it is a great pleasure for me to extend to you all a very warm welcome, especially for the speakers who accepted the invitation. Both conferences succeed in bringing distinguished speakers from 7 different countries. They are from Indonesia, Cambodia, Japan, France, South Korea, Malaysia, and Thailand. Also, the participants, which consist of lecturer, teacher, and student from various institutions. Hopefully, these conferences can be a starting point for the future collaboration among the teachers, lecturers, and the students from those various institutions. Moreover, I expect this conference can provide valuable knowledge to all the participants, and the outcomes of these conferences can be implemented by the participation for a better world. I also would like to express my sincere appreciation to all committee members who work tirelessly to ensure the success of the conference. And as Director of WNM, I'll open both of this conference. The second international conference on life science and biology education, with the theme Life Science and Biology Education, and the first Chemistry International Conference with the theme Enriching Chemistry Teaching Learning to Produce International Qualified Graduates. Officially open by saying Bismillahirrahmanirrahim.
Bismillahirrahmanirrahim.
And let me close by wishing you a very enjoyable discussion. Terima kasih. Wassalamualaikum warahmatullahi wabarakatuh. Selamat pagi.
Thank you very much for the Rector of Universitas Negeri Makassar, represented by Vice Rector for Academic Affairs, for officially open the first CIC and the second International Conference on Life Science and Biology Education. Ladies and gentlemen, before we close the opening ceremony, we will have a photo session with the Vice Rector for Academic Affairs and Dean of Faculty of Mathematics, and also the Head of Committee. So, and also with the keynote speakers and invited speakers. So we would like to invite all the keynote speakers to come to the stage for a photo session.
Checklist here. Photo session, Pak. Photo session, Pak. Photo session. Silakan. Masuk mata ini.
All right, that was the photo session. Thank you very much for all the invited speakers and also for the Vice Rector for Academic Affairs. Sorry, Professor, can we take again another photo session with the committee? Okay. So we would like to invite all the committee to come for photo session. We would like to once again invite all the committee member for the first CIC and also the second International Conference of Biology. All right, thank you very much once again for all the invited speakers and especially for the Vice Rector for Academic Affairs Universitas Negeri Makassar for attending our opening ceremony.
Tibrik atau langsung? We will have it 15 minutes.
All right, ladies and gentlemen, now we will have 15 minutes tea break before we start the keynote session. Thank you very much for your attention and wassalamualaikum warahmatullahi wabarakatuh. Once again, we would like to invite that now we are having 15 minutes tea break. And after 15 minutes, we will start the keynote session. Thank you very much.
perhatian untuk semua peserta. Sudah itu makalah. Nah, sekarang tuh waktunya istirahat. Kalian bisa atau bapak ibu bisa keluar untuk ambil kuenya, tapi tidak boleh dibawa masuk. 10 menit dari sekarang masuk kembali karena pemaparan pemateri itu akan dimulai dari Prof. Natalia. Ya, terima kasih.
Rasa sayang dilihat dari jauh. Rasa sayang dilihat dari jauh. Rasa sayang dari mana datangnya limpah, dari gunung turun ke kali. Dari mana datangnya cinta, dari mata turun ke hati. Sana gunung, di sini gunung, tengah-tengah bunga melati. Di sana bingung, di sini bingung, dua-dua saja. Rasa sayang dilihat dari jauh. Rasa sayang dilihat dari jauh. Rasa sayang
Bisa minta yang lain. Oke.
Invite dulu, saya
Ya, invite.
sebentar.
I would like to apologize for Dr. Hyunju because she has a little bit problem with the health, so maybe she will be a bit late for the keynote session.
Okay, great. Thank you so much for the Master of Ceremony. So for today's session, I will be the moderator for the keynote speaker. Before we are listen to the speech from the keynote speaker, let me introduce the speakers for today. For the first speaker, she is Professor Nathalie Bourguignon from France, and then her research interest is about marine biotechnology, seaweed, and algae. And her latest article that we can find in the internet is about identification of the quorum sensing signal of the opportunistic pathogen inducing bleaching disease in the red macroalga Halimenia floresci holobab. Yes, holobiont. Okay. And then the second speaker is Professor Kazuyuki Nobushi from Tokyo, Japan. And then his interest is about soil science and environmental science. And the recent article is about ecosystem carbon dioxide exchange and water use efficiency in a triple cropping rice paddy in the southern India, a 2-year field observation. And the last presenter, she is Dr. Hyun Chu. She is from Macquarie University, Australia, and her research interest is about STEM and mixed methods. And the latest article is Art Integrated STEM in Korean School. Okay, so for the first presenter, let me invite Professor Nathalie Bourguignon to deliver the speech.
Okay? Yes. All right, great. Thank you very much for this introduction. So, selamat pagi. Okay. Pagi. Thank you very much, Ruslan, for this invitation. So I am very happy to be here. Thank you for your welcome and your welcome of your team, amazing team of researcher, dancer, singer. Okay. And so I will present you a conference about seaweed. It is my specialty. Just before, I show you my position in geographic position in France. So the France is here, Malaysia here. So up and up to go to Paris, and after on the coastline of Atlantic Sea. So my laboratory is in Vannes, but your university has two main campus, Vannes, Lorient, and a short campus here. So we are near the ocean in Morbihan Gulf. It's like archipel of a lot of islands. So Nusantara, it's like archipel. So we are in the Gulf. We have 100 islands. So, and it's my campus of research. So to start the conference, I will show you different ideas about ocean. So we observe a remarkable diversity in ocean, 240,000 marine species have been described. Scientists estimate her number to be between 500,000 and over 10 millions. So fungi, bacteria, protists, microalgae, cyanobacteria, protozoa, and viruses. Most of them depend of rocky and coral substrates and sediment. So we observe a remarkable diversity. It's very important. With the existence of specific phyla in ocean. So this ocean of genetic, physiological, and biochemical diversity offers a privileged field of research and a large field of investigation for chemists of natural substances. So just to show you the diverse marine diversity and with specific phyla, we observe Echinoderma, Cnidaria, Bryozoaire, Acidia, sponge, and typical seaweeds. So seaweeds are extremely group of aquatic, eukaryotic, multicellular, photosynthetic organism. If it's low plants, no, it is no plants, no roots, no stems, no leaves, okay? And this group includes specific seaweed with a large size, different size, millimeter to, for example, 100 meter. So you have microalgae and macroalgae or seaweed. We have example of Pelagophycus spora, or the large Macrocystis example, and this Durvillae, it's amazing seaweed from Argentina. Example of Saccharina from aquaculture. So example also of Saccharina from France. So we observe 3 different groups with 2 different histories. So batic seaweeds are primary producer in marine ecosystem, food for many invertebrate and vertebrate, support for detritivores and decomposers. They are included in the structuration of aquatic ecosystems. The large forests in oceans, it's a kelp forest with brown seaweed, intertidal seaweeds on the rocky shore, and you could find also seaweeds in coral reef. So here you have some example of Littorina. It's herbivory, so it's food for this this organism. Here it's a kelp forest, And here it's Interdinal seaweeds on my rocky shore in Brittany. And here's the example of coral reef. So the important idea on this slide is to remember that these forests of brown seaweed are very, very important in the cycle of carbon on the planet. Sometime in your courses, you learn that terrestrial forests are very important for the climate change, etc., and ecological, but the marine brown seaweed forest is very, very important. And we estimate that the carbon sequestered by this algae are higher to the plant, to the angiosperm. So on this slide, it's just behind the algae, we find the human activity, and in particular, the woman. When the men went fishing, the woman harvested the seaweed. In Brittany, well, it's what a long time ago, but it is in the picture, it's a work of woman on Japara coast. It is a picture of my colleague Ita Widowati from Diponegoro University, and it is a collect of sargassum. And yesterday with Hoslan Jumadi, we and Fatma, we were looking for of the farmer who collect seaweeds in this village, Lekan village. And yesterday we were in Bati Murung Seaweed Factory for the transformation of seaweed. And so you could see again, woman. So actually we estimate around 32 million tons of seaweed by year are collected in the world, and mainly in China, Indonesia, Korea, and Philippines. And these seaweed are come from aquaculture, okay? 96% from aquaculture, and the 4% concerns wild seaweed. It is this figure propose the use of seaweed. So the first use is the commercialization of texturon, so hydrocolloid from seaweed. After it's human food, and in the last part concern cosmetic, chemistry, or fertilizer. World seaweed, and you can observe here the increase of the value of seaweed in the market worldwide. So the first application is as a jelly texturon. So you have the in food application. So you have the example with red seaweed, Eucoma capaphycus from your country, or Chondrus crispus from Brittany, and the extraction of carrageenan used as a jelly. Again, we can observe the extraction of agar-agar from red seaweed again, with the example of Gelidium or Gracilaria, and the use in microbiology or in food. And the last example is the extraction of alginate from laminaria or macrocystis or sargassum, and the use in human health or in dye. So it's hydrocolloid, so texturon, and it means it's a cell wall polysaccharide extracted from seaweed. The second application, I think you know that, it's a food application. So you could find in the world a lot of application as vegetable, is salad of wakame, for example, colerpas, maki, soup. You have, I don't know, crispy with a chili and salad. And here you have some example of French example. You can include polysaccharide in beer, in mustard, in marmalade, in soup. And the last application is the food application, cosmetic, thalassotherapy, pharmacy and medicine, or fertilizer. So, the distribution of seaweed on the rocky shore depends on geoclimatic conditions and various biotic as fouling, epiphytism, microorganisms, grazing, competition, and abiotic parameter, example, the solar radiation, hydrodynamism, temperature, salinity. So this, in response to this stress, because these parameters are stress, because it is benthic seaweed, so in response to this stress, various metabolites are produced to get adaptive or acclimation abilities, okay? And in my lab, we are not ecologists, but we are looking for this compound of defense, okay? And we chose since 10 years ago, around 10 years ago, we chose to work with invasive seaweed. Why? Because with this invasive seaweed, we have a lot of biomass. So we can work at industrial level, okay? And second, this invasive seaweed are nuisance for tourism and responsible for financial losses by research operators. So if we find solution with the discovery of new compound in this invasive seaweed, it could be great for the planet. So our objective is to explore this biomass for the research of new compound. And second objective is to work with eco-friendly processes to extract new compound, because water is very costly and solvents sometimes are very toxic. So we chose to work with environmentally friendly extraction method to have a high efficiency and reduce solvent and water and the time consumption. You could find our publication on the website if you are interested. So first, we start to work with cell wall, oligo, and polysaccharide. Remember, in seaweed, you have a typical cell wall with fibrillar part, with cellulose and hemicellulose, and charged part of this cell wall with sulfated polysaccharide, alginate, carrageenan, agar-agar, or ulvan. This polysaccharide have a lot of ecological properties. So I list these properties: structural and protective role. Example, give a flexibility on the cell wall. It is a barrier against external aggression, pathogen, grazer. So you can imagine if this cell wall have a lot of ecological protect function, it is because we have a lot of biological compound with activity, okay? So you know this molecule because they are in beer, in food, in your dentifrice, like clean for you, okay? And different spray, okay. So in my lab, we work with extraction-assisted, enzyme-assisted extraction. So we mix seaweed with commercial enzyme, okay, pluck, 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 and we obtain two extracts, supernatant extract, solid extract, and a residue. So in my lab, we test this solid extract against virus. So I show you who work against concerning herpes simplex virus. This. So here we test different extract obtained after enzyme-assisted extraction from invasive biomass. Okay, so example of Alimenia, Solaria, Ulva, Sargassum, and we evaluate the anti-herpetic, antiviral activity. And here you have on this line the antiviral activity, and in the center of this graphic, the high activity of the different compound. And we can observe that in red seaweed, in comparison with acyclovir, it's a common drugs, or fraction obtained after enzyme extraction, we obtain a very good activity. And here on this graphic, we evaluated the antiviral activity on virus resistance to acyclovir. And again, we obtain very good activity. So it's a work of Hugo of our lab. And the second graphic concerns the COVID. So we test again the same extract against SARS-2. And again, we observed that our extract inhibits intracellular replication of SARS-2 and reduce the production of virus in the supernatant of culture. So good application. So in my laboratory, we work also with phenolic compound. Phenolic compounds are typical compounds from brown seaweed with a large quantity of this compound. The phenolic compound, initial phenolic compound is a fluoroglycinol, and after polymerization, you can obtain different family of phenolic compound in different seaweeds. Again, this compound have ecological application for seaweed. For example, here on this graphic, you have the phenolic content of Pelvesia canaliculata during 1 year, winter, spring, summer, autumn. And you could observe that during the summer, you have an increase of this phenolic compound. So remember, during the summer, you have a high level of light, high level of temperature. You have a lot of microorganisms, a lot of invertebrates, so a lot of stress on the seaweed. So the phenolic compound increase. Here is the same. You have different seaweed here on the rocky shore. At the high shore, you observe Pelvesia canaliculata, here, Fucus spiralis, etc., etc. And so we evaluate the level of phenolic compound. And here again, you can observe at the middle of the shore, the high level of phenolic compound in seaweed. In the middle shore, you have a big pressure of the stress, okay? So this phenolic compound have antioxidant, anti-UV, and anti-fouling activity. So this phenolic compound find application in health, in human health. So you could buy different food complementary in the world. So again, in my lab, we test the extraction, the enzyme-assisted extraction with a phenolic compound. It was the PhD of Maya Puspita. It's PhD. It was a PhD student from Diponegoro University, and Maya Puspita showed that we can extract phenolic compound with this technology, and phenolic compound with high antioxidant activity. And in this work, we test a lot of enzyme, carbohydrates, or protease. In my lab, we work also with mycosporine-like amino acid. It is a small molecule present in a red seaweed, and again, it is ecological molecule in response with the light, and we can observe the relationship between mycosporine and the deep in the ocean. And this molecule have anti-UV and antioxidant activity. So, for example, in Grassalariopsis, it is a red seaweed. If you put this seaweed in raceway during 7 days in free light intensity, we can observe when we increase the light, we can observe the increase of mycosporine, and example, the polytinol mycosporine. And you could find this typical molecule in cosmetics against the sunscreen. So in my lab, we work again about the research of this molecule. So Hugo, in collaboration with Mexico, Sylvain from Mérida in Mexico, we are looking for the mycosporine in aquaculture with rhodymenia in different conditions of light stress, and we observe different mycosporine in this seaweed. And in collaboration with Gilson, it's a French company, we extract the mycosporine using centrifugal partition chromatography. And again, we can separate the different family of mycosporine. So we try to work also with seaweed as fertilizer. On this figure, you can see the different application of seaweed compounds in agriculture. And on the market, you can find different organic liquid rich in macro and micro elements, in betaine, in alginate, grow regulator. And we can observe the stimulation of fruits, the stimulation of grow. So, and they are active against pathogens. So a lot of application in agriculture. So in my lab again, we work again with enzyme extraction, and we evaluate different extracts from Solaria cordalis. Here it's a composition of this extract on this pathogen. It's very pathogen. It's Yoidium. It's a pathogen in French, in tomato. And here you could observe the efficacy of our product against this pathogen. And again, with Solaria cordalis, always it's an invasive seaweed. We test the soluble extract on the radish seeds, and we observe the increase of the weight, length of the sending. We observe also the stimulation of the chlorophyll rate. Okay. And with the solid residue, we study the bioabsorption capacity of the residue for different metal ions. And in conclusion, we show that the production of the novel compounds of fertilizer of bioabsorption, it's good solution for waste water treatment. And here it's for publication in collaboration with Poland University. And the last example is the example of the research who we've developed with Hostland. So Hostland came in my laboratory during 3 months, and he came with sargassum, invasive seaweeds, and he prepared different enzyme extracts, and he tests in collaboration with Yasser and the team of Hostland the different extract on the, it was, I remember, on the fertilizer, okay? It was with corn, huh? Corn, okay. So in conclusion, so you can see we have some possibility in French laboratory to work on the seaweed. And remember, Indonesia is a large, beautiful country with a large coastal line. And you are the main contributor of the growth of the seaweed in the world. And we can observe a very good biodiversity with seaweed biodiversity. Actually, you are leader in the culture of Caphaficus, Phocoma, Gracilaria, so in the production of carrageenan and agar-agar, used as texturant. So I think it's with French in Indonesia laboratory, we can find good connection because we can develop together a high project with seaweed. So thank you very much for your attention. And on this slide, you have different picture of my team, my French team, collaboration with Hostland, Hockey, and Ita Widowati. So, and here's the young doctor and researcher, because they are the motor, the center of the research is you and PhD student, master student. Thank you very much.
Okay, yes, that's fine, totally fine. Okay, thank you so much, Professor Bourguignon, for the speech. And yeah, we have listened to the comprehensive story about seaweed. And now we are moving to the second speaker. Yeah, please welcome for the Professor Inobusi, and he will deliver about soil microbial biomass and greenhouse gas flux in agriculture soil.
I should access. Just a moment.
Oslan, perhaps you need the same email with Zoom.
Zoom you sent me.
I stopped my connection with Zoom. I will inform you. So French. It's okay. Why understand? Why? One thousand. One hundred. Why hundred? Oh, 100. Yes, 100. Yes, and here around the
140 is yeah. The capacity is about 150.
okay. So she has this morning. Normally for the breakfast we eat together. But she said no, she don't explain something, but she so I think perhaps she had a lot of she need to prepare some baggage, I don't know.
But it's related to the yeah. See.
Because it's the dish.
Yes, she have consumed something allergic.
Last time when I was during the PhD of defense of fire, one day I was I drink smoothie with a lot of ice during this night. But yet, for this no problem. Yeah. I drink some tea with ice. Yes. Before my conference, I organize it here because it's sometimes we have program. But like the same thing.
You have been traveling to Jakarta. Because you say the Onegoro University is from Jakarta, right?
Yes. You have Jakarta, Diponegoro, and here Jakarta. No. No. It is.
Been traveling to Jakarta.
So this
this night I go to Jakarta.
Yes.
Semarang. I don't know. So
Semarang is Central Java.
yeah, and so I live in Jakarta, Semarang, Yogyakarta. Where in Yogyakarta? Yogyakarta. Yeah, okay. And I hope I and it was my first time. My dream is to see next time the typical house in your island.
With
yes, the eight hour by car. It's like this. One night. Okay. Yes. So next time I will visit Tohaj. I get so quickly.
Sorry to wait for a long time. Assalamu alaikum. Thank you very much for the invitation, and now I'm very happy to talk about algae again. After the French talk, I will present about Japanese algae and also Malaysian algae, Indonesian algae, all over the world algae. It's related to the soil microbial, the activity, including biomass, and also some relation to greenhouse gas emission in tropical and agricultural soil. My co-author is my new MS student of Tokyo University of Agriculture, Miss Kawashima, and the former Brazilian student, Silvio Ushiwata, also knows, and also my colleague, Dr. Oshima, Dr. Kato, and Dr. Yashima of Chiba University also. What is next? Okay, so the history of the microorganisms in the terrestrial ecosystem is long history. Since the Earth evolved and life came up about 40 billion years ago, and also the marine ecosystem become more critical in the situation, and also the salinity level increase. At that time, chemoautotroph already existed in the marine ecosystem. But the salinity is very high, and some of the pioneer microorganisms come up to the soil, the terrestrial ecosystem. That is not soil is not existed, but later on, some of the pioneer microorganisms like cyanobacteria or the other leguminous plant comes up to survive in the marine situation into the terrestrial ecosystem. And at this time, cyanobacteria comes out to become dry tolerance, similar to salinity tolerance. So once life comes up to the land, it's very dry situation. So it has some protection to avoid dry condition. So Bruggenalgae or cyanobacteria have some mechanism how to tolerate in the system. And the terrestrial ecosystem become much wider area in the many lands in the Atlantic and also the many continents. So increase salinity level, enhance the life comes up to the land. That is the theory. And also due to photosynthesis of the cyanobacteria, more effectively on the land surface to compare with the deep bottom of the ocean, more sunlight comes out. That's why once terrestrial ecosystem develop, oxygen increase up to here. So even before the marine system, some oxygen is creating, but cyanobacteria enhance oxygen creating and also help to establish terrestrial ecosystem. Then other terrestrial plants are supported by the mycorrhiza, another fungi, to absorb more water from the root. This is a present picture in the south end of the Chiba Prefecture, Tateyama, to find in the dry condition like that one, massive green mass. But once it stopped raining, it becomes desiccated and dry condition. But if you can put on the agar plate with BG11, nitrogen free, but some phosphorus and the other nutrients are supplied that can grow up like the very green, tiny mass. If you observe in the microscope, you can see some of the continuations of cell, green cell, and the little brighter cell we call heterocyst. What is a heterocyst? Heterocyst can fix nitrogen, dinitrogen from the atmosphere using the energy from photosynthesis. So it's the many cells separating, but once it's combinating, that they are separating photosynthesis or nitrogen fixation, working together. So it's no longer the single cell anymore. It's start to divide the function of the CO2 fixation and nitrogen fixation. So it's a kind of the very pioneer the microorganisms in the Earth. First evolved in marine ecosystem, then coming up to terrestrial ecosystem. You can see also some the shell. It's not so easy to see, but some the protection of the polysaccharide to avoid the dry up desiccation. You can make the liquid culture by using BG11. BG means blue-green algae. BG11 without nitrogen. But if you bubble in the clean air with some CO2, you can see the very beautiful cell culture. And I want to show you, Natalia, that one Japanese the columnist and scientist, Dr. Aramata, eating nostoc cyanobacteria with soy sauce. So even some of the cyanobacteria can be edible. In the southern part of the Kyushu and Kumamoto, it's very famous under the local food, which would be somehow the jelly material. Let me continue. If you consider the salt tolerance, like the desiccation, salinity level increase from 0 molar to 0.1 or 0.2 molar of sodium chloride. Usually, if without nostoc, the plant growth suppressed. But if you apply it with cyanobacteria, like this one, the root may be protected by cyanobacteria, and growth is better with nostoc cyanobacteria. So that is somehow the combination of the cyanobacteria with the nostoc cyanobacteria and the plant root. And salinity stress. So that can be some of evidence for moderating salt effect. We have some setup of outdoor experiment by using the non-stock the cyanobacteria, applying the soil surface to see the dry up and also the salinity level by using the moisture sensor, pH sensor, thermal sensor, and we continue like this one to compare with control without no stock. Now you can see some no stock as a dry condition, but if you apply the water or maybe under rain, the algae might be expanded. And sorry, in the level is not clear, but at the beginning in the August, in the very hot and dry condition in Japan, the moisture level is initially very wet, but after 1 week, it started to dry up. But if you apply it with algae, the blue algae, like the green bar, the moisture might be kept inside the soil. So that means that the no-stock application may help water condition better than the control. So the no-stock could delay the soil surface evaporation, like to cover the soil surface. Also, the temperature increase and very hot in summer, even in Tokyo. Sometimes the soil surface become 40 degrees over. But you can see the daily up and down. If you consider the without the no-stock, sometimes exceed 40 or 45 degrees in the midday. But if you apply the soil with no-stock, that can be moderate, not exceed 40 degrees. So that can be some chance for the supporting plant growth, even the very hot, dry summer, if you apply no-stock on the soil surface. So no-stock can reduce the soil temperature fluctuation day and night. So this is too much detail, but we can also check the soil chemical physical properties, and some data indicate the soluble carbon or soluble nitrogen can be accumulated once you apply the no-stock in the, as you can see around this value. Then we can also try to use this no-stock application into the highly dry up condition, very poor nutrient. If you apply no-stock, they can support plant growth, like supporting water or moderate the high temperature. So that can be rich nutrient to be the more diversity soil. Also, that is also some possibility in the future. We can also do some experiment in the Petri dish level without no-stock or with no-stock to compare with BG11 with nitrogen or without with nitrogen as a additional or without nitrogen free condition. Now you can see if you, even the water may support some germination, but it may stop. But if you apply the no-stock on the surface, that can support plant growth like this one. Even the BG11 without nitrogen, the blue-green algae no-stock can fix nitrogen. So that could be similar to nitrogen application system. So let me move to Uzbekistan. Where is Uzbekistan? Assalamu alaikum. That is also Muslim country. So I should learn also the Muslim culture in the Uzbekistan. And Karakoram, Pakistan is very close to Aral Sea. Aral Sea was the fourth biggest lake in the world, but it became shrink into only 10% in the due to the climate change. Because this is because the cotton growth area may use the fresh water before enter into RRC. So RRC in water inlet become almost dry up. So, and also the cotton field, they applied more irrigation to wash out the soil, salty soil. But the water may go down, but later it comes up. So salt may be accumulated in the soil surface like that one. So soil become sometimes whitish, not snow condition, but the salt, it's very salty. So there are some cyanobacteria might be existed. So actually the situation in 1977, that was the fourth biggest lake next to the Caspian Sea. But year 2014, it become dry up. Only the remaining part is like this one. All other the whitish, salty lake anymore. And the boat was already on the land. The salt is washed out by reaching by the cotton field. But the excess reaching causes increase of salinity due to the insufficient reaching with saline water, and irrigation system is insufficient. So river water decrease. So reaching enhance the salinity problem, and desertification increase the desert. So sustainable desalinity technology should be established instead of reaching. That is a biological chance. So cyanobacteria is pioneer back to about 25 to 30 million years ago, and fixed nitrogen and dry tolerant, salinity tolerant, and also the utilizes sodium and calcium and reduces the soil salinity. That may be the good health in the bioremediation. Cyanobacteria can help the plant growth. So that is a possibility. And maybe in the future, if you can get the CO2 from the fossil fuel, but it can be fixed by the cyanobacteria together with nitrogen, and sometime food or sometime the medicine. And the problem of soil, salinity soil, become healthy soil if you apply the top with some polysaccharide to build up the ecosystem, enhance succession, and more sustainable irrigation water system. We could isolate not only Uzbekistan, but also Nigeria in Africa, and Inner Mongolia, or even Vietnam, and sometimes mangrove, but sometimes dry condition on the footpath. So isolation site is all over the world. And sometimes even the Makassar in the Indonesia, Malaysia, we have also the tropical cyanobacteria isolated already. Now in the testing photosynthetic ability, nitrogen fixing ability, salt tolerance, dry tolerance, and excrete polysaccharide ability. We can keep some stocks isolates in Japan. We have some published paper and in 2013 by the energy people, Dr. Togashi, and also recently we have also the case study in Uzbekistan to isolate and utilize the desalination like this one. If you have interest, have a look. And maybe in the future, not only the Earth, but also the Mars or Moon. It's also dry condition, no oxygen. So we are working with NASA together with JAXA to see the tolerance of the cyanobacteria. Maybe in the cosmo agriculture might be possible to utilize the cyanobacteria. That might be still dream, but it's become getting some reality like this one. And back to the another problem is heavy metal. My colleague, Dr. Phil Brooks, he is working in China after retirement from England. He was working with Steve McGrath, who was also the famous Rosenstedt soil scientist. He is studying, they are studying the heavy metal problem, and by using the blue-green algae to monitor the survival of the salinity stress and heavy metal stress. If you apply with heavy metal containing sewage sludge, sometimes the suppression of blue-green algae, even the soil surface, but there are some the tolerant isolate comes up. That can be the maybe possibility candidate for the detoxification, not only salinity, but also heavy metal contamination. So the survival of cyanobacteria in the soil surface where sewage sludge the compost has applied. And the background, as I mentioned, cyanobacteria application could be promising in the desiccation. And also application of cyanobacteria could reduce evaporation. But there's some possibility to survive in the heavy metal contaminated soil, such as the sewage sludge application. So relationship between both the survival and growth of terrestrial ecosystem can be also the challenge of the new topic. So accelerated paper said sewage application, even they stopped 20 years ago, they realized heavy metal might be a problem. But once you apply the heavy metal in the soil, that remained in 20 years or more. How many students are younger than 20 years? Nobody. But if you are at about 20 years ago, you cannot remember what's happened. But the people knows this soil has received the heavy metal. Not only the people, but the cyanobacteria knows some remaining heavy metal back to 20 years ago when applied. So don't contaminate heavy metal in the soil. Please be careful. But cyanobacteria can monitor such system. And also in Japan, in Chiba University, one of my former PhD student, the master student, Mr. Omura, tested also the similar way in the heavy metal containing sewage sludge to compare with control. And this summer, we went to investigate the same site. It was very difficult to identify because more trees comes up around 20 years. But we could identify the place, and sorry in Japanese, but we collected the soil samples there and testing the physical chemical properties, heavy metal content, and as well as microbial response by ATP and chlorophyll content. The data is too small to show you, but the carbon might be still remaining in the surface and soil subsurface. And here is the heavy metal content. To compare with control in the blue bar, both surface and subsurface is some containing, but that is background, maybe due to the volcanic activity. But 20 years ago, once we applied the heavy metal containing sewage sludge, even 20 years later, still higher zinc or copper can be found in there. So even heavy rain every year, 20 years continuously, but there's still heavy metal remaining. And we also same situation, taking soil sample, put in the petri dish under the lightning to see the cyanobacteria or bioclast can be also observed in some way. And interestingly, ATP content in the as an indicator of microbial biomass. You can see the surface is higher than bottom subsurface in the control. But surprisingly, you can see if you apply the heavy metal containing sludge, ATP is still more or less half, which is an indication of the heavy metal effect still remaining even 20 years later. So in the future direction, sorry in Japanese, but we can continue the study to see the mechanism and also to find the heavy metal tolerant microorganisms and the moderation of the cyanobacteria would be possible. And finally, the talk would be in Malaysia, in the Cameron Highland, and also the Putra, Malaysia, and also the Kuantan in the other side. We could get some isolation in even the humid, the tropical condition by using the BG11. How much time remains? Almost. Okay, so I will conclude quickly, sorry, in Japanese. The survival in the humid climate, still possible to isolate the cyanobacteria and can be also utilized together with the empty fruits bunch from the oil palm factory as the moderation. So once you landed from Japan, it's a paddy field, completely changed to oil palm plantation in Malaysia. So the situation is completely different. But some microorganisms would be same as in Japan and Malaysia in tropical condition. And we can also presented my master student in the ESAFS meeting in the last August, the poster session to show some preliminary result. So students would be very encouraging to come and go nowadays after COVID-19. And we do some investigation and taking soil sample in Cameron Highland, similar to Marino in the Indonesia. We can see some the lavish and empty fruits bunch distributed. We see some the greenhouse gas emission, and we can see some the CO2, methane, nitrous oxide emission. But we are just restarting the investigation in Malaysia, and I hope in Indonesia, the Makassar people could help some future collaboration. In summary, cyanobacteria as a pioneer of the ecosystem and found in saline soil and arid region. Salt tolerance cyanobacteria can be used for the bioremediation. Heavy metal may affect the cyanobacterial activity as a very sensitive indicator, but somehow tolerance maybe. The survival in humid climate to be studied, not only the Japan, but also Indonesia, Malaysia. Thank you very much. Terima kasih banyak.
Thank you so much, Professor Inobushi, for the speech. And now, actually, there is still one presenter that are Hyen-Chu from Australia, but unfortunately, seems like she's having something with the health, so she is sick. So we, okay. Actually, she's coming. So yeah, we are waiting for her. Yeah. Please have a seat. So we are waiting for her, and actually from the presentation of Professor Inobushi, it's really interesting for me because some years ago, I have watched a Hollywood movie. It's about life on Mars. Yeah, actually, like there are some researcher, and then one researcher had left behind due to the rocket things, and then he managed to live on Mars after some experiments because they broke some plants as well, and then their colleagues can also bring him back to the Earth. So probably cyanobacteria can be part of the movie. Yeah. And then, okay, while we are waiting for the third presenter, probably all of the participants, because actually we are in the two mode for today, for the offline, and also we are connected to the Zoom meeting. So I know some of the participants already have some questions related to the papers, to the speech from the two presenters, but keep it for the next session. So yeah, as you can see from the screen, we are having around more than 100 participants as well from the Zoom meeting. So yeah, I'll give some chance to the participants here and also from the Zoom. If you are having some comments, some questions regarding the cyanobacteria or seaweed, and then for the third presenter related to the STEM area, so you can keep and then ask it later for the asking and question and answer session. Okay, sure. While we are waiting for the answer, please, sir, if you want to raise some question.
You're talking about the nostoc and cyanobacteria you experiment in Japanese. So my question is, you're talking so much about effects to the soil. What about the unfertile soils? Could we increase the fertile if I applied to the unfertile soil? I'm calling breeders, so many experiments in the field, but sometimes I found the unfertile soils. What about the nostoc? Okay, the second question is, you experiment in Japanese situations in the subtropic area. If I try in South Sulawesi, it's the zone of tropical zones. What about the effect? It's the same in Japanese situation or in the tropical zone, like in the South Sulawesi. So the last question, it's the nostoc and cyanobacteria, it's organic or unorganic or the fertilizations. And how much recommended to apply for the 1 hectare, for instance, for the corn I'm planting in the unfertile soil? How must be applied and when to be applied the stadiums of the plant? Thank you.
Thank you so much, sir, for the questions. So actually we are having 3 questions and then.
Thank you very much for the questions. And I'm very happy to have the question. And but I try my best to maybe second question to compare the Japanese and the Sulawesi tropical condition. I think that the Japan has also very hot and humid summer, but it's limited only June, July, August, September. So maybe limited 3 months or 4 months. But in Indonesia or many other tropical countries, it's whole year the hot. But nevertheless, in monsoon Asia, sometimes dry and wet circulation. So the microorganisms, especially the soil surface, may affect not only the temperature, but also the humidity and rain. So some cyanobacteria can survive even the dry summer. I have also experienced in the northeast Thailand, in Khon Kaen, that also very dry summer, and all paddies dry up. But still, I could find some cyanobacteria. Even the dry up, it's very difficult. No more green, but a black spot. But that can have a very high salt tolerance. Even in the humid countries, during the dry season, they can survive even without water. So the potential ability to the survival would be very great. And I cannot remember the first and third.
Okay, the first question about the effect for the infertile soil.
Yeah.
That's
Yes, I think so. Especially the low nutrient to compare without the nutrient. If you apply some phosphorus, for example, can accelerate nitrogen fixation. They need not nitrogen because they can fix the dinitrogen, N2, but they need some phosphorus. So if we, some little nutrient to support nitrogen fixation, phosphorus would be very important key factor. Okay. And the third question.
okay. And the last question is about, is it organic or inorganic? Yes.
Oh, the microorganism itself is organic. So yeah, but maybe the very primitive stage, maybe some inorganic, the function would be important. Okay, but such as the lightning for the fixed nitrogen would be also possible. But the major activity would be organic, the biological activity. Thank you very much for your question.
Thank you so much for the answer and also for the question. Okay. Now we are having for the third presenter, Dr. Hein Chu. Yeah. Okay. Okay, so please welcome Dr. Hein Chu, and she's going to present about our integrated STEM in science classrooms, STEAM program integration and practice. So Dr. Hai is from Macquarie University, Australia, and then he's part of the School of Education. And her interest is about STEM and also mixed methods. Okay, while we are waiting for the presentation, probably majority of the participants are teacher or student in the teaching university here. So the third presenter probably more be related to the teacher because it's related to the STEM, how to implement STEM in the classroom, how to activate the students for during the teaching and learning process. So you can provide us some questions to her related to the STEM or any thing related to the education. It is actually it's summer here.
Check.
Okay, I think it's ready. So please welcome Dr. Hyun Chu for the paper about Earth Integrated STEM in Science Classrooms, Team Program Integration and Practice. Time is yours.
Sorry for preparation. I was so passionate to share my studies with you. Is my sound okay? Yeah. And I prepared too many slides, so I'm not sure how to finish everything. My research is about STEM education in science classrooms. I will demonstrate how we did research with the school teachers and also researchers, and then how we implemented in school. Here is my overview. I will introduce about STEM project and then talk about the differences between STEM and STEAM. I think everyone here are familiar with the STEAM and STEM, right? And then I will introduce the theoretical framework and instructional models that we used for our program development. Also, I will demonstrate one program implementation as an example, and then make a conclusion. However, there are second program implementation that we are still under development. You will have an opportunity to participate in our research. So this project was based on district funding. It's all about developing innovative teaching approach and then investigating the impact of that innovative teaching approach program. And last grant is for help teachers and students science capital from this developed program. All these funds coming from Australian government, Australia-Korea Foundation. This one really emphasizes close interaction between Australia and Korea. Also, I had some support by Macquarie University. Okay, so it has been for a long time. I was involved with this program. I was chief investigator. Sonia Martin, Professor Sonia Martin will have give you a talk today, was my co-chief investigator in Korean side. And there are more professors, but I just selected someone who are collaborating right now with me. And these three professors from Korea, and these three professors from Australia. So we worked together. I also had many 2 to 3 Australia Korean school teachers and our researchers in Australia and Korea. So without this research team, I could not bring all my research outcomes to you to share together. As you know that STEAM has been with us for 5 to 7 years. And there are lots of research papers since 2018. You can find more than 23,600 those kind of publications from Google Scholar. We also had in Asia Pacific Science Education Journal. It will be explained about this journal by Professor Sonia later. The STEAM special issue in 2021. And those papers were downloaded more than, I think now it's more than 6,000, more than 7,000 times. However, we could not withdraw the one concept about STEAM. STEAM education is theorized differently depending on countries and researchers. So here, this is the general definition. STEM is a multidisciplinary approach, and it was implemented in the classroom like a silo subject, separate subject. But we talk about STEM research, STEM approach. STEM education is more integrated approach, and then the aim of STEM education for students learning science more in a holistic way. So it has to be always integrated approach. And we think that those STEM education can encourage students who may feel STEM is so difficult to them. So STEM can be one of way equity of a STEM approach in science classroom. We can include more students who like science or not. Those students have an opportunity to think scientifically, solving the problems scientifically. So let's start the definition of art. I didn't say this first part. STEAM is an arts integrated approach to learning that uses science, technology, mathematics contents as starting points. So science is the starting point, and we start to look at how arts can be integrated. In our research team, arts means here, it can be defined as any event or product that reflects a community's social, cultural practices and values. Example, painting, architecture, literature, history, even leisure activities and festivals. We consider those things as arts when we develop the STEAM program. The STEAM program was designed from these three perspectives. First, we really promote intercultural communication and science learning. So pilot project started from Australia and Korea. I am still working on this Australia and Korea projects. But in the future, we want to extend our approach. Maybe it can be Japan and Australia or Indonesia and Australia. And we employ engaging pedagogy. Everyone knows here about inquiry cycle using 5E learning model. We also use the 6E learning model. I will explain it later. And the multiple representation of knowledge and the knowledge enactment. Knowledge enactment is connected to 6E learning model. Because of intercultural components, from 2016, we really focused on integrating technology. After COVID, the technology emphasis was more there in our program. So we used online platform, collaborative online tools for students to work together from long distance from Korea and Australia together. Here is the STEAM program we developed. Since 2016, we developed about, I think, more than 7 modules. Everything starting from science. So module 1 starting from science concept light propagation, light reflection. We integrated the arts related to festival. Sydney has vivid light festival. Korea has lots of media facade during Christmas time and some special occasions. So we used this light festival to learn about the physics concept about light. Seasonal change, this is one of the example I brought here. I know you are all biology teachers, but I made lots of photos, videos. So maybe if I can demonstrate this one, you can understand me better. And you can apply the approach when you develop your program or when you design your lessons. There is wetlands. Wetlands, we focused on historical place. Korea has wetlands around DMZ area between South Korea and North Korea. So we used that area to talk about biodiversity in wetland. Also, Australia has very interesting place around the wetlands. So these kind of historical perspectives were integrated when they talk about wetlands. All those 4 programs, when we implemented, we shared it with Australia, Korea teachers in 2016. And then currently, we are working on those projects. First, around our school, you will have opportunity to participate our research related to forest around our school. It's connected to biology, but we are still under developing this program. That's why I could not bring it here today. And we completed Zero Energy House project. This one concept is connected to heat transfer, energy efficiency, but more connected to traditional house and architecture. I delivered the lecture yesterday. And climate change and One Planet school project. This is club after school project. We are still under developing. Our programs next round will be based on NOAA technology, embedding Arduino or microbit for collecting massive data, and students can be somehow, because science, we don't teach coding, but these Arduinos and microbit are all connected to coding. And then we will use like video making and data science. Our direction is more towards integrating technology into STEAM program, but for students to understand our science, integrating arts there. Okay, so all our programs, including under development, was shared with the Korean teachers. So we worked together really hard. Then every time when we have products, we always share the Korean teachers and Australian teachers. And we also just completed our symposium with the Australian teachers. So I was collating everything since 2016 to now, some good outcomes to share with you. Here is a theoretical framework and instructional model we use for program development. Our program, all the program is based on constructivist theory. And it's to help student knowledge construction rather than teacher transmit all the knowledge. And we emphasize the students' small group work, collaboration. And we think because knowledge is socially and culturally constructed, students may hold multiple perspectives about the same issues. And we expect those approach will increase student positive perceptions and attitudes towards learning. All based on this constructivist theory and the situated theory, based on all these theories, including inquiry theory. Now I am going to introduce instructional model. This is 5E instructional model that helps teachers to implement inquiry approach. As you know that 5E learning model is well matched with constructivist theories, but our focus is integrating arts into every 5E stage. Engagement stage, they start to know about what is today's the module's situation. So for example, student, I will show the further examples in my next slides. Most of the time, they explore science knowledge connected to their cultural exploration at the engagement stage. After they find out some science knowledge here by science hands-on activity, then they design or create some artifacts. And these artifacts has to be elaborated using the science concept they explored here. And what they prepared will be presented. So it's at the evaluation stage. The presentation will get peers' comments and feedback. So we followed this 5E learning model, and those explanations were published in this paper. So if you want to know how we followed this 5E learning model for STEAM development, you can find further information from this paper. And we also followed the 6E model because current years, we start to embed socio-scientific issues into STEAM approach. So when we include the socio-scientific issues, the topics like wetland, forest around our schools, climate change, you can see those topics has the opportunity to include socio-scientific issues. So those socio-scientific issues, we really emphasize student enactment. So they learn science, then they have to use the knowledge to do something. If they found out my area has some problems related to wetland, maybe they start to destroy the wetland, make the wetland very polluted. And they have to do something like make a poster and present the poster at the city council. All this enactment is at the last stage. So we started from 5E learning model, but the program moving towards more socio-scientific issues. Now we added 6E, it's enactment, enact stage. So enact stage here, it means it helps students put into action the values and practice they explored from the science classroom. And also it helps the student understand the connection between science knowledge and practice, as well as the values of science knowledge. As you know, there's in Korea, at the TIMSS and PISA test score, student score is at the top group, but they don't see learning science, the value. Their value recognition is very low at the bottom, and their attitudes towards learning is at the bottom. I feel it's very strange, right? Their knowledge here, but attitude is here. But it's similar in Australia. Australia, the PISA score is above average. It's really good, but student attitudes towards learning is below average. So we really think that if we include the social scientific issues into STEAM, but STEAM is more inquiry, more guided inquiry, so they can see the values of learning science and they can use the knowledge to practice, to do something about my community. So it helps the student to develop global citizenship. It's because why we are moving this direction, you know that scientific literacy is developing all the time. First vision of scientific literacy really focused on scientific knowledge and process. So when we open our curriculum, it talks about what kind of knowledge we needed to cover, what kind of scientific process students needed to apply when they do those problem solvings. But vision 2, it's connected to STS, you know, right? Science, technology, and society. So now science educators start to insist about connecting science into everyday life, like the first round program. It's really connecting arts, student every life, student culture, events, everything into science. Vision 3, it really emphasizes global citizenship, critical scientific literacy. It means students have to learn knowledge and they have to use the knowledge to practice something. So now the scientific literacy moving towards Vision 1, Vision 2, Vision 3, because of those literature studies and other research studies. And we start to move, including more social scientific issues into STEAM program. So here is example of a program implementation. Yesterday, I gave you some example about, I think some of you were at the lecture, the Zero Energy House. Today, I decided to bring seasonal change. So seasonal change and night and day, this teaching was based on 5E learning model, but we also include one more things for research purpose. My initial focus was to investigate student concept development. So we included model-based learning. So here, we choose this topic because Christmas is in summer in Australia, but in Korea it's a snowy winter. So when they discover about it, they start to see easily different seasonal changes in each country, different seasons at the same year of the time. And also because of this very direct observation, they become very engaged about seasonal change lessons, season lessons. And it helps Australia students study the season, the science together, like a collaborative learning. This was how we prepared lessons. lesson 1 and 2, it's at the engagement stage. They prepared some kind of artwork to show their nature, to show their life. Lesson 2, they introduce their artwork together and then propose research questions, some problems, some questions. Lesson 3, they don't study together but separate. In the science classroom, they start to do some science activity to solve the problem they raised at the lesson 3. Lesson 5 and 6, it's evaluation stage. They share what they found and then get feedback and then revise again and then present again. So this is piece of student artwork from Australia. It's a student painting, but there are a lot of things. Can you find it? The trees don't have a bark. It's Australia, one of a eucalyptus tree, and it loses all the bark. And see, there are green. I am from Sydney area. There are lots of green area. But look at the soil. The color is unusual for Korean students. So when they look at this painting, Korean students start to ask lots of questions when they meet the Australian student. Of course, at this stage, we don't expect any science explanation. It's just showing my nature, my life. Korean students, they decided to take some video. I don't know why. Korean students love to make video clip. So they made a video clip and then they showed their life. Maybe I can show you just a little bit. Sorry, no sound, it's okay. We will look at the mobile phone. So his mom waited him at home and she couldn't talk to her son and made a text message. And then he said to his mom, "Don't worry, I will play." And the video continue how he plays around. So it goes going to show amusement park and everything. So even though this video it's just about everyday life, but Australian students will start to see, oh, it's the same of the year. And at that time, I think it's June. And Korea is green, Australia is still green, but we are moving. It's cold in Australia, no snowing, but cold. So we are all wearing jacket, but Korean students are all wearing short sleeves. So they start to ask questions from the video. Australian students start to see there are lots of buildings, no forest, not many green things. So this is a good starting for them to talk about season, day, and everything. So they make their artwork. Now it's time them to meet together. So when they meet together, they introduce each other from the Sky meeting. It's 2016. None of us were familiar with the Sky meeting. I mean, I was foreign scholar, so we used the Skype all the time, but it's different in school. So we had a hard time. Anyway, now it's much easier to do this kind of class teaching using Zoom. So here is conversation. Is there snow in Sydney in July or August? And Australian student answered, not in the city, but in the mountain area somewhere else. And then the question is like a Korean student asked, what time is it now? And then Australian student just shows their like watch. No, we cannot see clearly. More closely. Then bring this big watch. This is small things. In the classroom, we can just say Korea is 7 p.m. Korea is 1 p.m. and Australia is 3 p.m. We can just say like this, right? But when they hear and when they see some information through this kind of international interaction, students are very motivated to ask questions. Why do we have a different time? Why do we have different seasons? So anyway, we have to teach the seasonal change, how the sun and those earths, how sun revolve around axis, everything. But they can do all this learning more actively with fun. Okay, so now, what is the stage? They raised their questions about season and time, and they moved to science lessons. During science lesson, we adopted this. How do I remove this one? Okay. We adopted this model-based learning cycle. So students make their initial model, individual model, before teacher teaches something, to do some science something. They start to draw why we have different seasons. Based on student response, teacher prepares some intervention here, then they revise their initial model. After that, they decide this mid-model and have a short presentation. Teacher find out student conceptual problems. And teacher provide again instruction, intervention. Then student will work together as a group. So see here, this part is personal model. When student had their personal model, now they can have a discussion, negotiation with their group members, and they can decide their group model. When they go through model 1, model 2, model 3, at the end of the model, I think in the classroom, at least 2 groups among 5 groups were able to present textbook type of appropriate science model. So teacher is really prepare everything. Teacher is busy to find out what's their conceptual problems, but there is not much teacher's direct teaching. There are points teacher have to do some demonstration and showing some video clips, but it's all based on student in models. So here we found out when students draw their initial model, they always emphasize distance between Earth and Sun. And they are confused about Earth's rotation, revolution, and they talk about change of Sun a lot. Then teacher did some intervention and moves here. So let's see what teacher has prepared. Torch. And then all those things for students to fix their conceptual problems. After that, they collate their mid-concept again. Then teacher find out student has difficulties about sun's altitude. And now and also about the axis. So now teacher prepared all those things. And this is a student model. You can see at the beginning, there is Korean words, so you cannot read, but from the drawing, you can directly see. Initial model, it's really based on how much light arrives in on the Earth. But later, they talk about those revolution, and they draw this S6, and they even talk about what's happening in Korea in in Australia. So they start to apply what they explored here for Korean case and Australian case. So here is my teacher. Okay, let's listen to her comments and then I will move close. I cannot hear you. Okay. We cannot hear her voice, but she said STEAM program was really first time for her to implement, but it was okay for her. The lesson was not heavy for her. It was only 5 to 6 lessons. And she was able to integrate more things around these 6 lessons. It means she brought some English books, talk about different seasons and some paintings. So she thought it was good for students to learn science actively with fun. So here, my questions was student engagement and conceptual change. And we found out that the STEAM program helps students to pose questions, helps students to explore the response, and they can generate their explanations together. We really think this socio-cultural context make the class a bit different, small things, but it's lots of preparation for researchers and teachers. But now we prepared all the theoretical framework, instructional model. It's a matter of how you can integrate into the classroom. And should I stop here? Or can I introduce? Yeah, okay. This is our current study, and we are interested in integrating SSI into STEAM. You know that it's very powerful and helps students to see the science. And instead of 6E, we added INET. And this is a program example about our forest. We included turpentine ironbark forest in Sydney area, but the forest student can explore anywhere around the school, right? And we really want students to use systems thinking. And this forest topic can work with teachers in other subjects like geography and history and social sciences. This is our module we developed. We are now developing worksheet and some PowerPoint slides for teachers. And this one, module 1 is abiotic vectors. Module 2 is about field trip. They go to forest field trip. And module 3, data analysis, data collection. And they collect data from module 2. Module 3 is data analysis. And module 4 is more INET stage. And these modules I just skipped. And at the enactment stage, students start to collate the piece of knowledge they explored from all the modules to make a magazine, to make video clips, and then they post it to like school website or city website, all those activities. This is what we are doing. We want to look at systems thinking. Systems thinking is important. It's what Australian curriculum is emphasizing. Systems thinking means it's not that new. It really emphasizes interrelation, interdependent, and interacting. So you can see that there are interrelated area like plants, hydrology, and then also geology, this kind of geology. And interdependence means it's connected to food web, they are interdependent, right? and also interacting, those things influence each other. This is only from science. You can start to add more things from society and human influence, how human can influence on this system. So system is kind of key, we think, for STEAM education. STEAM looking at different fields of knowledge for student problem solving. And this is one of concept map to show someone's systems thinking. So this was drawn by scientists. That's why when the person is talking about biogeochemical flow of carbon dioxide, the person includes a lot of science categories, like ocean interaction, land interaction. You can see it's really huge and complicated. But most of the time, biogeochemical flow of carbon dioxide, we can find those kind of simpler concept map at student level, year 10 or university students. So here is our forest module again. And this is the concept map my pre-service teacher draw in 10 minutes. So it's not well really developed, but the teacher is showing kind of lake, rainfall, flooding, and it's also connected to influence, has some relationship with the ironbark forest. And this is another teacher's example. You can see this time, they also talk about flooding and then how it's connected to economic perspectives. So here is activity. I will upload this PowerPoint. Pfizer will upload send you these PowerPoint slides. Please draw your concept map around the science and then send it to me. You can draw it in Indonesia. Dr. Pfizer will help us. So I want to see how you can extend your systems map connected to science and all around like human influence, political, social, economic perspectives. And because of that, we are also preparing pre-service teachers. Our course is related to STEAM context. We developed a questionnaire to investigate pre-service science teachers' systems thinking in STEAM context. Here is the link. We need your participation to calculate the reliability. So it's 57 items. While you are answering to this item, you will learn what's the meaning of systems thinking in STEAM context. What kinds of ability STEAM teachers should have in their mind. So our own research, it started from researchers' confusion. We really didn't know how to integrate arts, how to have this kind of systems thinking. We struggled for a long time. So I think this is our research and practice. I want you to participate in our research too. Thank you.
Okay, thank you so much, Dr. Hai, for the really marvelous presentation. So you can have a seat, and then we will continue for the Q&A session. I know that probably there are already a lot of questions related to these three presenters, but unfortunately, due to the time schedule, we can only have two questions. One question from the offline and also one question from the online mode. Okay. Yes. Okay. Please, the man with the
Do, teacher. Sure.
Thank you. Good afternoon, Ms. Bogonion, Mr. Inwasi, and Ms. Uncio. Okay, Muhammad Unsashala from Biology of UNM, this university. I'll ask you about, first is for Mr. Inwasi about the cyanobacter application in soil. We know this application is so advantageous, but there are some harmful toxification in microbial activity in soil. So how we tackle this problem? Because we know that cyanobacteria is tend to overblooming, and some species in cyanobacteria is toxic for the other species. And the question for Ms. Uncio is about STEAM. People in class is, some people like me is identify us as introverted person, and the extroverted person tends to dominate the inquiry session in class. How a teacher give the person in class with the introverted characteristic to get the chance to make the inquiry more suitable for them. And some people, even in biology, have identified themselves as no art sense, with STEAM that is approached with art. How we collaborate with them without hinder the people with good art sense. Thank you.
Thank you so much for the question. So, okay, first, Dr. Hi.
Yeah. Hi, I have to check your question again. So first you asked how you can help students. Yeah, okay. Yes, that's a good question. We did a study with Dr. Sonia and Dr. Pfizer about student engagement, verbal and non-verbal student engagement. We found out that many students were very quiet in Korea, especially in Australia. Some students are quiet, but it doesn't mean that they don't think. They don't follow you. So you really have to carefully observe them, their eye movement, and when they like show some smile and about your teaching. So for their active engagement, I often create online sharing form like a Google document, or Padlet for collaborative online comments and feedback. Some quiet students actually actively show their engagement there. So I think we have to really observe them carefully, which student is quiet, but how they are following you. I was very quiet in the classroom, but I studied very hard. So, I mean, teacher has to really observe it.
And upsets in STEAM approaching.
The people with no art sense, like no drawing skills and etc. How we give them STEAM approaching, I mean.
So I think I could not explain the program very well. Arts does not mean that music and drawing only. It's more liberal arts. So we open for culture and human influence and socio cultural practices. So when we include this one for their explanation about, for example, Light Festival, I think they didn't have that fear about arts sense, because what we are evaluating is not the artistic work piece. It's about, I mean, they could draw like a round shape and they could insist this is a sunflower. It's okay. But if they explain their idea using science concepts nicely, that's the key point. And we have to understand science drawing is different from artistic drawing. So when I try to collect student drawing in science classroom, at the beginning of a science classroom, I emphasize what's the artistic drawing and what's the science drawing. So that kinds of introduction, maybe teacher needed to provide it to students.
I think it's crystal clear. So we are moving for the Professor Enobuchi about the question about the toxicity. Yes.
Thank you for the questions. And then the succession is not very simple way. So the pioneer come up, and sometimes the successor may come later to utilize the pioneer's the footprint, like the substrate or the dead body, to be the food for the next generation. So succession takes time, and actually it maybe develop the diversity. So there are many different microorganisms to utilize the different ways. So I think the biodiversity is now a very hot issue, but it's also the long history to come up from the beginning until the full the richness of the world nowadays. So I think it's not short story to explain. And also the soil itself is also quite diverse, from wetland to dry land. And also microorganisms living there may tolerant to the wet condition or maybe tolerant to the dry condition. So diversity of soil may also reflect to the biodiversity of soil microorganisms. So I want to show some slides, but unfortunately, battery has run out. Same as our stomach, maybe. We need more energy. But anyway, thank you very much. Is that answer to your question? Thank you very much.
Thank you so much for the explanation. Yeah, I'm really sorry. I should apologize for the online session from the Zoom. I couldn't make it for the question, but as you already received the book of abstract, it's including the email, the contact for these incredible presenters. So if you are having questions, comments, or probably a project that you can do together, I think they are widely open to be contacted with. Yes. Okay, I think this is the end of this session for the keynote speaker. Thank you so much for the remarkable presenters. So we already listened from 3 continents, from Australia, from Asia, and Europe. And I think we already gained a lot of knowledge and information. So I think I will return to the Master of Ceremony.
All right, thank you very much. Please give a big applause for all our keynote speakers. Thank you very much, professors, for the really insightful session today. And also thank you for our moderator for guiding this session. Please give a big applause for our moderator also. All right, ladies and gentlemen, now we are going to give a certificate of appreciation for our keynote speakers. So we would like to invite the head of committee, Dr. Wiharto, Associate Professor Wiharto, to come to the stage and give the certificate of appreciation for all our keynote speakers. So for the first certificate, we will give to Professor Nathalie Bourgonon from University de Bretagne Sud, France. For the second is for Professor Kazuyuki Inubushi, and the third is for Dr. Heian Chu. Ladies and gentlemen, please give a round of applause for Professor Nathalie Bourgonon, France. Thank you very much, Professor, for sharing your insight during this conference. And the second, we would like to invite Professor Kazuyuki Inubushi from Tokyo University of Agriculture, Japan. Thank you very much, Professor, for coming to the Faculty of Mathematics and Natural Sciences, Universitas Negeri Makassar. Thank you very much. Ladies and gentlemen, the next is, we would like to invite Dr. Heian Chu from Macquarie University, Australia. Please give us a round of applause. Thank you very much. And beside the certificate of appreciation, we will also give a souvenir. So here is souvenir from University of Makassar. So this is the building, the main building of Universitas Negeri Makassar. And we also have a traditional fabric. We call it batik from Makassar, and we would like to give it to all the keynote speakers today. Okay. Okay. So please. Yes. Yeah. Oke, langsung sesi di sini. Maju Kika. Maju Kika Bulan. Kita datang lagi terus. Maju lagi kita. All right. And for the traditional fabric, all the keynote speakers will directly use the traditional fabric. So the first is for Professor Nathalie.
Sekarang. Okay.
And the next is for Professor Kazuyuki Inobushi. Kazuyuki Inobushi. I'm sorry.
Oh. Is that too big? Okay. Okay.
And the next is for Dr. Hayan Chu from Macquarie University, Australia. All our keynote speakers looks really nice using the traditional clothes, batik from Makassar. Thank you very much, professors. Thank you for this really insightful session. And ladies and gentlemen, that was the end of our keynote session today. Thank you very much for your active participations during the session. Wassalamualaikum warahmatullahi wabarakatuh.
30 bikin lagi mereka.
And for your information, we will continue for the second session of our conference today. We will start the invited speaker session at 1:30 p.m. Makassar time. Jadi kami informasikan untuk sesi invited speaker, kita akan mulai insya Allah pukul 1.30 waktu Makassar. Jadi mohon untuk peserta yang bergabung secara online untuk menyesuaikan. Terima kasih. Wassalamualaikum warahmatullahi wabarakatuh.
Yeah.
Untuk para pemakalah, kami silahkan untuk mengambil konsumsi di ruangan. Di depan lift ada ruangan, di panitia sudah menyediakan konsumsi.
Sekitar 5 menit lagi kita mulai.
Oh iya, iya.
Alright, Assalamualaikum warahmatullahi wabarakatuh. Ladies and gentlemen, good afternoon and welcome to the invited speaker session in the Faculty of Mathematics and Natural Sciences, Universitas Negeri Makassar. Ladies and gentlemen, now we will have our invited speaker session. For the first is Professor Fahrul, and also we'll be continue with Professor Oslan Jumadi. So I would like to invite the invited speakers to come to the stage. Please give a round of applause for our invited speakers. All right, ladies and gentlemen, for the first session, Professor Fahrul will explain about his research regarding exploring potential of halophilic bacterium isolated from hypersaline environment from bioremediation. He is from Department of Bioscience, Faculty of Science, University of Technology, Malaysia. All right, without further ado, I would like to invite Professor Fahrul to give the presentation. The time is yours.
Terima kasih. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi. It is a great pleasure to be here this afternoon. After how many years? After nearly 3 years, we undergo a process, we call it lockdown. And then within that 3 years, we always had online meeting. And once I got an email from Professor Mushawir that we will have something like offline. So I was so excited on this. So I would like to thank all the organizing committee of ICOLIP number 2 to allow me to be present this afternoon to share my experience in the research. I've been in academics for nearly 27 years, and I still feel a lot of things to learn. There are so many new technology that has been out in the literature that we have to keep up. Sometimes I say to myself, I have to stop after some time because I cannot keep up with the recent knowledge, especially the knowledge in bioinformatics. which is very demanding. This something like you need to know some mathematical equations so that you can simulate the biological activities in this particular computing program. So I'm not used to this kind of very emerging technology, but I try my best to encourage my students, especially the student from 2022 and above, to get involved with many types of research. And then since we have lockdown, so that's make our research become very competitive in terms of to get a research grant. At the same time, we have to also to compete with many things. Then we have to embark somewhere so that our KPI, key performance index, annually will not be neglected. So we still maintain number of papers to be published, number of student, PhD student, especially to be graduated within the stipulated time frame. So here I am. So thanks so much, the organizing committee, for allowing me to share my experience. But this is one of the activity that I did during my sabbatical year. I've been something like administrator for the faculty for nearly 10 years. So suddenly my faculty was dissolved at that time. From biosciences department become from the faculty, biosciences faculty become department, so that I'm no longer part of the faculty. So they changed me to the Faculty of Science. So I got something like invitation to do my sabbatical in Turkey. So I take this opportunity to do this research. So it's like you wanted to explore. Exploring is a big word actually, but my interest is actually on the halogenase enzyme, looking at the halophilic bacterium. So in the laboratory, we try to compare between lab techniques and isolation from lab techniques isolation work in the lab, and also the molecular approach. So basically using molecular but ecological approach, we can study the diverse of microorganisms that are present in the polluted environments. So many are phylogenetically different from pollutant degrading strains that studied in labs. That will show that if you study in situ, direct from the source, because you must understand that this microbe, when you bring that microbe to the laboratory, you cannot actually grow them all because in the lab system you have some limitation, because you of course you will learn microbiology that the bacteria need something like suitable pH and then suitable not only temperature and so on, so that in the environment is much more suitable. So you can study about the whole population of the bacteria in the polluted system. Whereas if you bring some soil sample or water sample to the laboratory, you might be able to isolate certain microorganisms. So that is different between the two systems that you wanted to study. So this molecular ecological approach cannot establish the mechanism capable of doing pollutant degradation. So that's the limitation. Of course, you will know from A to Z type of microbes present, but of course you cannot characterize them individual. So that's another disadvantage of studying molecular ecological approach. But there is other advantage as well. But some of these microbes occur in response to pollution, and they have significant influences on the fate of pollutants, because some chemicals, the structure is very complex, so it needs several microbes to carry out the degradation process. So in the environment, multiple species of microbes share ecological niche. Among these, one population best adapt to this ecosystem, so they are become predominant. As others, we consider that as minorities. The molecular ecological method detect dominant species And then whereas the traditional method like isolation techniques, methods fail to do that kind of things. And then I always compare with, between lab culture, natural ecosystem. So it is totally different in terms of nutrient concentration that you provide in the lab. Normally, the media for cultivation of pollutant degrading microbes has higher concentration. So the target pollutant and nutrients compared to the ecosystem. We must appreciate in the environment because certain microbes that present in the environment, they are able to degrade very low concentration of pollutants. So that suggests that they can values or the enzyme is so much high affinity towards this substrate. So that's the special about this in the environment. Whereas you put in the lab, that's become something else. This resulting in, if you grow them in the lab, the enrichment of fast-growing minority population will happen. So the microorganisms utilize various cooperative interactions with other species for their growth in the ecosystem, which include syntrophic and so on. So what happened in the environment? We have a network of carbon sharing of pollutant degrading bacterial consortium, differences in the physiological conditions to presume that lab strain can also efficiently degrade pollutants in the environment as they do in the lab, suggesting the importance of analyzing in situ activity. So that's why sometimes we need to consider this from the environment source. So there are many examples for molecular approach. Some to be very specific. Sometimes you amplify the eDNA sample from PCR, followed by DNA sequencing and so on. So these are examples, many molecular approach that has been published in the literature. The molecular biology techniques, it can be used to study the abundance, the diversity, activity, and functional potential of indigenous microbial communities in various metrics like water, soil, air, waste, and so on. So it's like for different microbial communities that can be studied. I mean, I'm sure you already in your class, you study about the genome, DNA genome, that you can obtain as much as you can the genetic information. So some of these bacteria that we can easily do the full genome sequence. Then you keep that in the database, for example. And then we look at the RNA things like transcriptome. And then you study about the proteins. There are many proteins that you wanted to study. Study of proteomics. All these terms is, as I said, in the first hour of my lecture this afternoon, is like you have to keep up with this proteomic, metabolomics, and so on. So this is totally different when you do your PhD in 1990s. You don't see all this terminology. You must need to keep up with all this. Skip high throughput sequencing. Nowadays, we talk about this high throughput sequencing. When it was first introduced about 5 to 6 years ago, 7 years ago, they are very expensive. But nowadays you can do it. But still, some labs, they are not able to do it because they still say that it is very costly and very complex. Therefore, it's not to be a diagnosis tool. It's just like when you have some special project, then you can do it. So this technique able to identify the new taxons, new function of on microbial communities, and so on. So these techniques nowadays you can get the service from the lab, from the public lab that offer this kind of metagenomic sequencing, and also the full genome sequence as well. So these are examples of something like proteomic studies. You can study about the 2-digit electrophoresis. You identify what are the important proteins, and then you can get the protein sequence and so on. So I just the microbial biome, I just skip this one. So what is important when you do metagenomic? So recently, apart from doing pollutant degradation, I also study about the metagenome of honey. Because nowadays the honey is like some people may say that the honey is not genuine. It is a sugar compound. but how we wanted to analyze it. Sometimes you need to go to the source as a control. If you declare that the honey is really from the jungles of a certain part of your country, then you need to know what are the species that are present in the honey sample, for example. So this is you can determine using this technique, metagenome study, to study the full metagenome of the honey, lebah, nisan lebah, things like that. So this is very powerful techniques nowadays that people normally use this to even to show that how authentic your honey, for example, because of this superfood. Mass sequencing amplicons. So this is just to show that mass sequencing, you wanted to sequence the whole genome of each individual microorganism that are present in your sample. So my interest is basically to look at the xenobiotics. So everybody knows, I think, the xenobiotics, something like highly contaminated. It can be chemicals, pesticide. So these artificial compound are present in our environment. So this need to be treated. Normally, to treat it, it is very expensive process. We just rely on the original microorganism to do this. So as an introduction, so I just basically we received a grant from Turkey that time, because in Turkey they have a soda lake, they have a hypersaline lake, and so on. So we were assigned that time between 2018-19 and 2020 to analyze what are the possible microbes that are present in Lake Tuz, one of the Turkey lake. So these are example, a model substrate that we use in the laboratory, like 2,2-dichloropropylene acid, DL2-chloropropylene acid, 3-chloropropylene acid, monochloroacetate, and so on. So these are the only example of a model substrate that we use to test whether microorganism that present in a hypersaline environment can degrade this type of chemicals. So I mean, the basic principle of degradation is like most of the toxic chemicals, you have halides group. So the idea is that you wanted to cleave the carbon halides structure. So for example, you have 2,2-dichloro, you have two fluoride ions present at carbon number 2. So you wanted to remove both halides so that you have substrate. We call it, you have to transform from 2,2-dichloro become pyruvic acid. So pyruvate is basically most of the carbon source for the bacteria. So this kind of bacteria is a special bacteria that present in environment, because E. coli cannot produce this kind of enzyme, we call it dehalogenase, whereas certain organism, it can produce a dehalogenase enzyme to degrade this compound. Another one is DL2-chloropropylene acid, the position of halide at carbon number 2 as well, but whether it is a dextro or levo position. So it can be both positions, either one. So this dehalogenase character is like, it is stereospecific, suggests that they can act on carbon L position or D position. So maybe some dehalogenase they can act on both positions. So we call it non-stereospecific. So this is the character of this kind of enzyme actually. So having that kind of idea, so we traveled to Turkey. Because you can see from this what you call it a map from the sky, from the satellite, you can see that in this Lake Tuz, a lot of salts present. You can see very white area compared to the blue area. So they will say that water is very limited. So a lot of salt there, 70% is salt used in Turkey. But during winter, that's the only time that we had to be able to culture our, to take the sample during winter time. I'm not the one who went there, it was so cold. So other people go there and pick up the sample, the water sample, and bring into the lab and we do the full genome sequence. I mean the idea is to check what are the possible microorganisms that are present in this kind of environment. Just to be very quick, the problem statement is like very simple problem statement to have the idea that that there are many kinds of environmental conditions that some microbes might be able to degrade this kind of pollutants. So there's no study about the lactose, seeing the scholarship from the Turkey. So we just wanted to study the microbial population. And then the genome analysis, we carry out the genome analysis of bacterium that we managed to isolate from this lake. I mean, there are many objectives. There are 4 objectives, but I don't want to elaborate too much about it. You can check the papers and so on, the published work that here, just to share with some of the students. To see the significant research, to see the bacterial diversity using metagenomic approach, and then to discover dehydrogenase fluorescent bacteria from saline habitat that can remove pollutants, and then to data on gene related to metabolic activities. So that's the last significant of studies. And the pro research, I just keep very quick. So lactose is actually what we do. We wanted to get to do the genomic studies on the far left-hand side to extract the DNA, and then isolation of dehydrogenase fluorescent bacteria. That's another strategy that we do. And then, of course, to study the full metagenome, of course, is part of the whole project. Result is discussion to perform metagenomic investigation on the microbial population of lactose. So basically, we have ranges of microbes that are present, and then of course we need to check the physicochemical of the water. It is high contents of salt, and the pH is above 7. And then we have a range of microorganisms present from bacillus to bacteria the bacteria that never you come across in your mind. But among these microorganisms, some are a lot, some are abundant, they call it. In terms of the phyla, is many compared to others. But of course, as I said, in the first place, you cannot really characterize what this microbe can do, but it is present. Sometimes, although it is small in small quantity, like bacillus, for example, is very small amount of bacteria that are present, relative abundance compared to others, but maybe the presence is very important in terms of protein degradation. But I'm not surprised to see this kind of data, because all I can say that looking at the geographical of lactose, there is no water coming in. The environment is clean. It's not really heavily polluted. So maybe the abundance mechanism suggests that there are many in there, but the function is maybe for something else. But for protein degradation, I cannot say there is none, but there is something happen in the system. So Firmicutes much abundance compared to others, and then upon the genus level, Rambouillet is one of them. So when I look at the literature, it's basically Firmicutes like gram-positive bacteria, normal flora, and so on. So, but we are still interested in the protein degradation, anyways. And then look at the Rambouillet also have been published, so that not surprised. We are not really shocked that we found Rambouillet, but of course in the literature there are some other studies also suggest the same organism are present in high salt condition that we make the researcher become happy at that time. Apart from that, we wanted also to study about this full metagenome function. Because not enough to know how many abundance of type of microbes, but we also need to screen what are the possible function of this bacteria to be present in this kind of environment. So the most, as I said, I'm not expert in this kind of PIC-RST2 software because it is, of course, you can get it from the you can download it in the internet, for example. Then you can always estimate what are the possible gene or enzyme are present. So based on this, we found that protein degrading enzymes are present So it suggests several pollutant degrading enzymes like 2-hydroxy dehalogenase. That's what we expected to see. And then we have DHA haloalkane dehalogenase also present, acetate dehalogenase also present. Apart from that, there are other enzymes also are present, like glutamate synthase and so on. They have their own function, especially the one that allow the bacteria to survive in high saline, because you know the osmotic pressure. So how to make this bacteria survive? They need to have some balance in terms of ions and so on. So the student to isolate the Encarata bacteria capable of degrading halogenated compound. So this, I mean, their job to do some isolation. This is totally different from in situ studies. So now you bring the sample to the lab. Of course, you need in the laboratory you do some enrichment studies. So you provide the substrate and you allow, assuming that these are the potential microbe taken from the source. So again. This very basic isolation techniques, you need to identify the bacteria, you grow the bacteria in the specific substrate, the selective media to allow this organism to grow in 2,2-dichloropropanilic acid, as I said, as a model substrate. And then you have, you identify certain microorganisms like bacillus, although you can see the abundance is very little compared to others, but still the function is very important. But we identify the microbe as Bacillus subtilis. Some Bacillus, we just name it a strain. Of course, it's difficult for you to really confirm the name of the genus of the species, but based on the similarity, of course, you estimate 100%. Yeah, I mean, when you do some blast studies, I mean, the quality assurance is very important. You don't know, people submit the sequences in the database. Some people, they really submit whatever they found, the sequence. But there are other tests that to confirm, maybe biochemical tests, pairing with this, what do you call it, this test of CCS, you can really identify the name of genus, species of the microbes. So that's we suggest this microbe belongs to Subtilis and Thuringiensis. Of course, you need to check how fast the cell can grow in the selective media that you provide, like 2,2-dichloro, and then you can see that this bacteria can grow faster, like 7 hours compared to others, like more than triple times. Then you check the halide release, how much chloride release, if the cell can grow in this selective media. Of course, you don't expect 100% release of halides because depend to the the Km values of the enzyme present in the bacterial cells. And then you check the pH as well, what the pH, possible pH, is the best pH for this bacteria to grow. And then you check on the temperature and salinity. Also, you check the best saline condition and so on. What I like most is like to characterize these proteins. So we amplify the dehalogenase using published primer sequence. The student managed to amplify one gene, bacteria, from one of the bacteria from strain H2 strain. Of course, when we do sequencing, it is not 100%. So it is not 100% match to the published sequence, but what that will be, although not 100% match, but we just proceed with the experiment. I mean, so based on the conclusion that we do, it's like I just to short my talk this afternoon. I don't want to go into too much details on the protein analysis. So what we do, we successfully profile the bacterial population and their abundant present in the lake as well as protein function. They provide information related to the adaptation, environmental potential, and then we isolated 2 bacteria strains like Subtilis and Thuringiensis. Then we also identify a potential gene that we managed to amplify, and then this agency could only amplify only in one of the bacteria, which is Thuringiensis. I mean, this is only the work that you can carry out, like sequencing, protein prediction, and so on. You know, make sequence analysis and so on. So that's so yeah. I mean, I don't want to extend more to allow my colleagues to continue his talk. So for more information, you can always WhatsApp, or you can contact me through emails. I can send you the full paper if you're interested to see what are the further work that has been carried out from this partial sharing session this afternoon. So thank you very much for this afternoon, for allowing me to share my previous sabbatical work in Turkey. And I leave the floor to you.
Thank you very much. Please give a round of applause for Professor Fahrul. That was a very detailed explanation regarding the haloalkaline degrading microorganism for bioremediation. That's very interesting. And ladies and gentlemen, I believe that you already have questions on your mind, but before question and answer session, we will have Professor Oslan Jumadi, who will present the role of organic fertilizer on greenhouse gases emissions and soil microbial from tropical soil. Please welcome Professor Oslan Jumadi.
Thank you very much, Mr. Citra. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Selamat siang, masih bisa mengikuti acara? Thank you. My great honor to standing here with, it's front of my supervisor actually, and then collaborator, Professor Inubusi and Professor Natali. Okay, today I will presenting our long journey research, mostly about fertilizer to reduce the greenhouse gases emission. And I'm focused today to present organic fertilizer on greenhouse gases emission and soil microbial from tropical soil. So I would like to start my presentation to give a question. Why we consume fertilizer and why we should manage? Okay, so if you see this very distinguished tomato with fertilizer and without fertilizer. So in agricultural practical, without fertilizer, the soil will be depleted and the plant will be particularly difficult to grow. So we need they need fertilizer. So yeah, this is another sample in the leaf for lettuce. If without fertilizer, particularly for the macro nutrient or macro fertilizer, nitrogen, phosphorus, potassium, and yeah, you can see the control is a good growth. Good example in tropical soil in the corn. So you can see the picture of the corn with a deficiency with some kind of fertilizer. So that's why the demand of fertilizer is increasing year by year, and of course that accelerated with the increase of population of human. Yeah, because human need food, all right, and then the intensification agriculture need one of the method is application of fertilizer. So no wondering this increase year by year. However, if you see this graphic, if we put nitrogen fertilizer example in the soil, so in the soil there is a process that can be uptaken by the plant for growth well, but some soil microorganisms also use it, and several process of this microbe, like nitrification that delivery by nitrifier, the nitrifier will produce metabolic secondary of that process. We call it greenhouse gases. You can see this N2O, nitrous oxide, and the methane by the processes of methanogenesis by the archaea methanogens. So if you put more, so it means that could be also increase the greenhouse gases. Okay, and then some pollutant, call it nitrate, NO3 minus, that will leach from the soil. Why? Because soil is also negative and nitrate also negative. That's easy to leach or going to the water body and make a bloom in water ecosystem. So why that's happened? Because the soil has capacity to buffering the nitrate. So if you put more, it means the plant nitrogen will lose more because there's a buffering. The plant cannot absorb all nutrient if access more. And there's a volatilization, denitrification, and increase of organic. Why increase of organic? Because microbe use also nitrogen or macronutrient for growth. So no wondering if you put some nitric fertilizer, that also increase the organic of the soil. Of course, we expected grain protein, forage nitrogen for feed the animal, and nitrogen straw for for also animal. So also increase. But the increasing of fertilizer to the soil by government panel for climate change said will increase also call it greenhouse gases. This is, you can say, this is a new report by IPCC, that's by UN, said that you can see the nitrous oxide, the methane, and carbon dioxide is this is methane, nitrous oxide is steady increase. The carbon dioxide is fluctuating because some uptake by the photosynthesis and some kind of disaster from can produce carbon dioxide. But anyway, greenhouse gases is increased year by year, and that estimated 23% of the total greenhouse gases is produced by agricultural sector by application of mostly fertilizer. Okay, so that's why the increase of these greenhouse gases also parallelly the IPCC monitor the increase of temperature of the earth is a come up, okay, year by year, and that is believed that caused by greenhouse gases. Why that's one? Because the greenhouse gases can absorb the infrared that goes to the atmosphere and then they send back to the earth. So the earth will go warm and the climate change will happen. So that's we concern of the greenhouse gases. That's why the leader of all countries in the world is joined together, that we call co-op conference of the party. All the leader of countries join together to combat, to reduce minus plus to steady state the temperature minus and plus. The last one is Dubai. I think that this every year the United Nations join together to make a policies how to maintenance the temperature, so the climate change we can reduce for the next generation. So for our scientists in agriculture sector, we have to participate for reduce these greenhouse gases. So the option that possible we can propose is, yeah, of course, because 23%. So we have to manage the fertilizer. Okay, so one of them is changing from synthetic fertilizer to organic one, as well as to inhibit that process, drive the nitrification. So we call it a nitrification inhibitor. The most potent is, of course, the synthetic one. Okay, from process of synthetic one that we call it DCD, diazidamide, change to the NIM example. Or we design fertilizer with slow release fertilizer. So this is several chemical fertilizer which can reduce the process of nitrification that produce greenhouse gases, but this is nitrification inhibitor is expensive, particularly for developing countries. Indonesia may be between that, but my expertise is DCD is not available in Indonesia. Even available is very expensive. It cannot applicated in practical in Indonesian farmer. So we have to find some solution, and several articles said that several plants can have a potential for inhibit this process. Okay, as a nitrification inhibitor. So Indonesia, after research, we have a plan that we call NIMBA or NIM, that several in India, Pakistan use it for insecticide, and some using also mixed with fertilizer. So this is have a spectrum to inhibit the process. So me and my former student that now work in Universitas Sulawesi Barat, before we are using molecular docking. This is a very challenging topic because we just download the activity component of some plant and then make it join with the enzyme that responsible for nitrification inhibitor. So like you see, this is some kind of active compound in the MIMBA. Okay, MIMBA, Azadirachta indica. Yeah, this plant is particularly grow in tropical countries, so it's very good. We thinking global and act locally. Okay, so several active compound, and we doing in a computer basis molecular docking analysis, at least 3 applications, and then we download active compound from a website, and then the enzyme that responsible oxidize, catalyze the process of nitrification that we call it ammonium monooxygenase, and yeah, nitrate reductase, there is 4, and we combine that one. So there is one active compound that is more energy. So this is more potent to combat nitrification. So this is the deoxyribonucleic complex that becomes strong binding energy for inhibit that process. So we expect that there is a slow oxidation ammonium to the nitrate. So we expected that plant will absorb more efficiently of nutrient. So we can get nutrient uptake efficiency and then less nitrous oxide. Between 30 years, we design experiment and with a collaborator with Chiba University, Britain, SAT, and to use organic organic fertilizer. So this we tested in Balit Serelia, Maros. We tested also in Sokolia, Goa, and in greenhouse biology farm. Testing about using a sample Azolla. Okay, so we use Azolla, floating form that mutualistic symbiotic with the cyanobacteria that in which sense I already talked that can fix nitrogen. And we use Mimba, okay, organic to inhibit nitrification, both from wetland is mean paddy soil, because paddy soil is a anaerobic condition. Okay, so the metagenesis will happen. The gas methane will mostly emitted compared to the aerobic soil. So we tested both kind of soil with comparable synthetic one and organic one. So we conducted in the Maros Balit Serelia, collaborating with Balit Serelia. We usually use a closed chamber technique, standard technique, and then of course the farmer don't worry about the fertilizer that we use. They're thinking about the production. Okay, even they say, oh, this is good for global warming. They don't know. They just expect the harvest. Okay, the harvest. So we also determine the growth of the plant. You can see this. Of course, the control is more scarce compared with the fertilizer one. So from this experiment, Professor Nubusi and Yasima Sensei also already reviewed 2021. Actually, this publication 2019, and you can see the total nitrous oxide. Okay, the total nitrous oxide from urea with neem. Urea with neem is Mimba, okay, organic, and zeolite. Zeolite is a slow release fertilizer. Urea, solid, and we combine again. And then we combine with DCD. This is synthetic one. You can see the DCD very, very potent, very good to reduce the nitrous oxide, but this is not applicable for Indonesian farmer because that's expensive and not available. Okay, so at least we can reduce around 50% of nitrous oxide by using organic nitrification inhibitor. I think this is one of challenging to participate to reduce the greenhouse gases from the soil. So you can see urea, solid, and neem 66.8% nitrous oxide. We can cut it. So also we can get somehow not the prices of the weight of COP. COP is the harvest. Okay, but sure the urea more better. The synthetic one more better. Seems if you put some nitrification inhibitor in fertilizer, the plant will also, there's an inhibition there. So this is a comparable of the corn. So we believe maybe farmer can accept it and can use it. Another option for organic, we replace it with Azolla. Okay, replace it of urea to Azolla. This is our work 2015. This is Azolla mostly blooming in rain season at now, and our student collected in the also Maros. Azolla, floating fern that symbiotic with cyanobacteria. Azolla pilata, you already know that, and then this most contain much nitrogen because the bacteria freely subsidy give the plant to grow more. So we collect it, and then at this time we make a compost, and then we realize, this is very easy to make a compost because it's already hygroscopic. And then we try in the pot experiment by Kangkung, and then we collect the water of leaching, how much. This is we conduct in the biology farm. And then we also doing an incubation experiment. And then at this time we collaborate with Chiba University with Professor Inobushi to analyze the nitrous oxide. This is also a very point. We have two greenhouse gases that can analyze that one. So this is the result. So you can see this table. Okay, nitrous oxide, urea around 4 times more higher than Azolla 1. You can see this one. This is urea. Okay, this is urea, and this is Azolla. Even higher Azolla we put in the soil, the nitrous oxide will less than urea. So ammonium changing, and also nitrate quite, because we compose it. So we thinking that the organic one to become ammonium, that process we call it ammonification. Okay, so when we compost it, so the protein will convert to the ammonium and goes to the nitrate. So we have much nitrate in this experiment, and then you can see the plant biomass is not significant. Even the control a little bit same also, but sure, urea more enhance the plant. And recently we are collaborating with Spain company that we call Tibrio. This company produce a kind of, we call it O protein for the feed aquaculture, or lipid, or fras, and chitosan. So I wonder if they were right or Oslan, but and our collaboration is they ask us for testing the fras, but I also thinking why only fras? Why only fras? So we thinking also to using this mealworm or insect to produce the protein. So we collaborate with Bala mealworm to cultivate this mealworm. This is something we call it ulat tepung or ulat Hongkong. Okay, so that's a fras. That's as organic. This is the waste when we cultivate breeding the insect. There is a waste. It's a waste from digesting. Okay, so we breed and then collect the fras. Even some of our students are thinking for candidate for entrepreneurs to sell it as fras, but it's still developing. So we try in the field experiment and incubation experiment. We use the fras. The fras, after we analysis, is not so much contain nitrogen. It's around 3 or 4% compared to urea around 45%. So as well as we conduct also soil bacteria metagenomic. Prof. Farul already talk about metagenomic. We also wondering the microbial change when we use this organic fertilizer. So we conducted in Sokolia farm, and same method as before. UPK fras, and okay, in incubation we can see the production of nitrous oxide, the urea UPK, this one, yes, this one first they higher and then slow down. But we wondering why the price after first week, second week they are growing. We thinking about the ammonification, and then ammonium will goes to nitrification, and there is also nitrate. And incubation for carbon dioxide is I think the graph very beautiful. We expected more carbon in the soil, more carbon dioxide. Okay, more organic, more carbon dioxide. At the end, after one month, we collect the soil and then we analyze metagenomic bacteria. So this is the control soil. Soil without fertilizer. You can see the community of bacteria seems no dominance, no there is no abundance there. But when we add the synthetic fertilizer, the Nitrospira, Nitrosomonas community, nitrifier community is a little bit increased. And surprisingly, but I think not surprisingly, the bacteria enhance. We're thinking about additional micronutrient will also enhance the diversity compared to the control. But when we add the fast, the acidobacteria will dominate. So there is a change, even between 1 week. That is very interesting. And then we're thinking that's maybe caused by production of nitrous oxide. And we plot it by ternary plot. You can see that phylum, the phylum acidobacteria with highest quantity in the first sample, around 70%, UPK 50 and 30%. So after we study the article, seems that the gut of the mealworm has contained dominant by the acidobacteria. So it's released by digestive thing. So this is comparable share microbe between urea control and the frost. You can see that the application of fertilizer synthetic more enhance the bacteria. And now we think to fit this it's not we fit the mealworm with the azolla. This is very primary experiment. Last one, we has collaborating with Professor Natali by using seaweed. This is a collaboration with University Britain Sat with under PSC Nusantara. So we collect the sample in Laikang as yesterday we went there. We collect Capapicus alvarezi and sargassum. So before I went to the France, we collect the sargassum and then we dry. And thank you, Natalie, for facilitating laboratory during my France. And we analyzing the composition of sargassum and Capapicus with some basic method, protein, sucrose, sulfate, polyphenol, uronic acid, as well as anti-radical activity, pigment. And the last one, when I back, I thinking to use the extraction to coating the corn. So this is the background of comparable Capapicus and sargassum. You can see the sugar in Capapicus more than sargassum. That is common because polysaccharide in Capapicus more than sargassum that contain alginate. Okay, so here we thinking because we use enzyme assistant extraction with 4 enzymes, and we thinking why we don't use bacteria to extract the sargassum. So we collect bacteria, we isolate bacteria from abalone because abalone eat the sargassum, and we thinking bacteria will degrade. So unfortunately, from 25, 15 bacteria, only 2 can degrade the organic one. My former student will explain this more detail in this seminar after this one. And by we can access that this sequencing belong to Bacillus subtilis and Klebsiella, can degrade CMC, and then we use for degrade the sargassum to gain the extract, and then coating the corn, seed corn, to more better. So under laboratory, you can see fermentation bacteria control and metalaxin. Metalaxin is a chemical to hide the insect when we planted the seed. Another experiment by extract algae by water, we tested in the field. Okay, this is a week after transplantation, the upper from plant height is we can see the brown extract algae, of course different with the control one. Even the comb, quite that when we fermented the algae to extract and then apply in the corn, will enhance the growth of the corn. As well as we analyze the methane from this field, because this is when the raining season, and then remarkable that algae with the fungi will increase the methane, but not so high for the common concentration. But also we surprised that the gas N2O, this is the red one. Red one is a nitrous oxide from the urea, and the below is from biostimulant that we extract from the seaweed. Is much lower than urea one. So the conclusion: this organic fertilizer has a potential to reduce N2O emission compared to the urea fertilizer, of course, and then the major process of production of greenhouse gases, process metagenesis, nitrification, and denitrification, directly through the release of greenhouse gases from carbon and nitrogen compound present in the organic fertilizer. And addition of organic fertilizers management can affect soil microbial community that carry out nitrogen and carbon cycling processes in the soil. So thank you very much for Ministry of Education, Culture, Research and Technology for support this work. We wish to thank our research team, nitrogen and carbon cycling in the soil. Thank you very much for listening. Thank you.
Thank you very much, Professor Oslan Jumadi. That's a really nice presentation, as we know that greenhouse gases is global warming is an important problem that we need to solve together. And one of the solutions that is proposed through the research of Professor Oslan is the use of organic fertilizer. All right, ladies and gentlemen, now we are at the question and answer session. So I would like to invite the participants who wants to ask questions. You may raise your hand. Okay, we have, please, we have one question. So maybe the committee.
Thank you, Imchi. So my name is Yasin Ikram from the University of Islam Makassar. For the first speaker, you told so much about the microorganisms in the biomolecular marker. you successful isolated of the bacterial strains in 2 kinds of the bacteria, if I am not wrong for listen to you presented. But my question is, how could be controlled? Because I'm plenty worked in the field, bacteria, fungi, and virus is so much attached to the plant. So I'm mice breeder. It's very, very difficult to control. So many farmers recommend if my plant bacteria comes or fungi comes, it better I put in than apply of the pesticide or other. So what do you recommend for control these the kinds of the organism? So you successful isolated, but my question, how could be controlled? The second speaker, Mr. Jumadi, in your slide, you presented the sum of experiments, nitrogen, synthetic, but still no in the market or in the town. I think still no farmer use it in new treatment. There are many, but unorganic nitrogens like urea, 44% in the one package. So if we give to the farmers 100 kg of the urea, they are inside 45 of the nitrogens, but unorganic. Your treatment is nitrogen inorganic. Do you have the already distribute all your product in the WNM? Maybe we could be try to my farmer. Can't. And you already collaborate with the Balai Cereal, Balai Penelitian Tanaman Jagung Cerealia. Yeah. It's very nice. You experiment in the greenhouse and to the field, but no characters you observe of the codes. How could we take the conclusions? You have 7 treatment, nitrogens, unorganic, and so on, T. I don't understand what is it. But that's very important. No data you observe from the experiment in the pot and in the field. So how could we take the conclusion of your experiment? You talked also metalaxy. Every time I use metalaxy, giving my corn to the farmer for control of Peronoschlerosis padamedis, bulai, donimeldo. No mice Indonesian release high variety for resistant to the Dinmoldo. Eventually Maros. No variety hybrid or OPV. All variety is very susceptible to the metalaxy. So how could be recommended for this situation? Thank you very much.
Thank you very much, Mr. Yasin. So Mr. Yasin have 2 interesting questions for the first speaker and the second speaker. So may I give the first speaker to answer the question? Please, Professor Fahrul.
Thank you very much, Professor, about your question. Actually, we are not controlling anything here. It's like we wanted to study the profile, the profile of the microorganisms that are present in this particular environment, and then from there, whether this microbe can do the job of pollutant degradation, especially in high salinity condition. Cuma kita nak buat profiling sahaja untuk memastikan ada tak sebarang mikroorganisma ataupun mikroba yang terdapat dalam dalam keadaan kepekatan garam yang tinggi yang boleh degrade selected substrate atau selected chemical compound. So kita jumpa, we found at least using metagenomic profiling, we can see that certain microbes abundance, certain phyla abundance banyak jika dibandingkan dengan phyla yang lain. Tetapi kita tidak boleh menentukan apakah keupayaan mixed culture tersebut, sehinggalah kita membuat isolasi dan kita mendapati ada 2 potential strain yang boleh menjalankan proses degradasi bahan toksik. Itu yang kita dapat Bacillus subtilis dan thuringiensis, strain H1 dan H2. Sebenarnya ada lagi extension of the investigation that I don't put into my slide to allow time to my colleagues to present. But you can always text me or WhatsApp ataupun email. There are papers that published related to the extension of the work. I thought I wanted to save this for the next ICOLIP. I'm just joking. So I just keep others. It's like for the studies, is that we managed to amplify the specific gene dehalogenase based on the metagenomic profiling in terms of the gene that are present, the proteins that are present. So we found this, we amplify using PCR. We found the halogenase, and then we do the full genome sequence after that. So based on PCR or a specific dehalogenase, we characterize based on bioinformatics. We do protein ligands, same as what Prof. Junaidi explained just now. We cannot test that in the laboratory because we managed to just amplify the gene. We have limited time to go to the lab that time during lockdown. So we just sequence we get the DNA sequence, we get the full protein sequence, and then we just do in silico analysis to model the protein structure, and then we do the protein ligands analysis. Of course, you can see the protein ligand interaction. That one we cannot really say that is the best substrate. Was based on the affinity between the substrate based on the the strength of the energy binding. That's not really accurate measurement to say that. To confirm, I can say it is best to go to the lab to test this protein, whether it can degrade the substrate, and then get the KM value. Then we know that this substrate is very high affinity towards this enzyme. But we just do during in silico investigation only to get some idea that this dehalogenase present in certain microbe that we found that in the high saline condition. Of course, you can study the number of acid amino present, what are the important acid amino in terms of that can work well in the salt. I can remember there are few acid amino good in the presence of high salts condition compared to the normal pH, things like that. So that's the further extension. And then we also do the full genome sequence of this microorganism, I think H1 strain. Based on the full genome sequence, it's actually more than one dehalogenases are present in this bacteria. In our initial study many years ago, started this job dehalogenase back in England, in Leicester University and Cardiff University. They found that only two dehalogenase in one organism, or maybe 3 dehalogenases in one organism. But our current study in 2022, we make a full genome sequence. We found about 9 dehalogenases are present. This is another curiosity. Why this organism bother to have this gene, 9 dehalogenases? In fact, you know that 1 or 2 dehalogenases that suggests non-stereospecific is just enough. But to maintain 9 kits or 9 genes is much more difficult compared to one. This goes towards fundamental studies about the regulation of these dehalogenases, things like that. So that's about more towards. I don't see how we wanted to apply this in the environment, but it's obviously based on fundamental investigation. Just self-interest at the moment, not yet to apply this. But of course, in the environment, of course, you can see there are many natural microorganisms that can degrade pollutants. So thank you very much. I hope that will clear, Professor. We are not to kill, or this is not. This I don't know whether it's pathogenic or not pathogenic, but this environmental organism. We study about the properties of the organism in high salinity. What in there in this? Apa yang terdapat dalam high saline? Sebab grant daripada luar, so we have to take sample in their exploration in their country, things like that.
Thank you very much. That was the answer from the first speaker, Professor Fahrul. Thank you very much. And we would like to invite Professor Oslan to answer the next question.
Okay, thank you. Terima kasih, Pak. Ya, good question. Kita tidak berusaha untuk menghilangkan urea. That is a research. So we compare urea as common that farmer use in Indonesia. Okay, we give another solution, another fertilizer, organic. So we have to do research. So after this research, yes, still the urea available in the market. Of course, the urea is subsidized by government until now, but we don't know for the next year, 5 years later. If not subsidized, is increased 10-15 times. So yeah, yes, and you get only 50,000. Okay, it's very cheap, but it's subsidized by government, and the money government from us also. And if the country still subsidized, means some economical, still developing. Okay, so we try to make an option. Okay, mengurangi dan sekaligus meningkatkan lingkungan hidup. How we maintain the environment by some kind of fertilizer that eco-friendly. Okay, that's our goal actually. It's still minor research. So this research is not solve all things that you think. So the metalaxyl, same background. We use metalaxyl. We compare with the extract from sargassum. So many others. But this is still in laboratory, only for the shoot. We not applicate in field already. But if we compare in the laboratory, because it's control, I mean we control it all thing, no soil, the water is sterilized sample, so of course we can get a better result. Okay, so yeah, we still develop for the research. Thank you very much. Okay.
Thank you very much for Professor Oslan. Please give a round of applause for all our speakers. All right, ladies and gentlemen, we believe that there's still a lot of questions, but we are now running out of time, Professor. So maybe we close the question and answer session. And of course, if you still have more questions, you are allowed to contact Professor Fahrul and also Professor Oslan for more questions. All right, ladies and gentlemen, that was the end of our invited speaker session. Thank you very much, Professor Fahrul, for explaining the potential of halophilic bacterium for bioremediation, and also for Professor Oslan for the potential of using organic fertilizer. We do hope that in the future it can be produced in a mass scale to give more benefit for the society. All right, ladies and gentlemen, for the closing, I would like to invite Vice Dean for Cooperation of the Faculty of Mathematics and Natural Sciences, Universitas Negeri Makassar, to give the certificate of appreciation for our invited speakers. So please welcome Ibu Dr. Andi Asmawati, MSc, and also we would like to invite the Secretary of Biology Department and also the study program, the head of study program, to come to the stage and also join the certificate, the appreciation for our invited speakers. Ladies and gentlemen, please give a round of applause for Professor Fahrul. This is the certificate of appreciation for Professor Fahrul. And this is the certificate of appreciation for Professor Oslan Jumadi. And of course, we also have a souvenir, which is the miniature of Universitas Negeri Makassar. Given by the Secretary of Biology Department, Ibu Dr. Rahmawati. And here we will also give to one of the most productive researcher in Biology Department, Professor Oslan Jumadi. And the last but not the least, this is the special fabric of Makassar with the traditional pattern as well. So we would like to invite Bapak Dr. Musawir Tayyeb to give this special clothes of Makassar. So this use the alphabet of Makassar and also the picture of Makassar hero, Sultan Hasanuddin. Yeah. It looks really nice and it suits Professor Fahrul very well. So as you can see in Professor Fahrul clothes, this is the picture of Makassar hero. This is Sultan Hasanuddin. Okay, and the next we will also give the traditional pattern batik for Professor Oslan Jumadi. As you can see in his clothes, it has a like alphabet. Also alphabet in Professor Fahrul, it is traditional South Sulawesi alphabet. For the next is photo session. Okay. All right, thank you very much, Professor Fahrul and Professor Oslan, also for Vice Dean for Cooperation and Development, for the Secretary of Biology Department, and also for Bapak Dr. Musawir Tayeb. Thank you very much. Ladies and gentlemen, that was the end of invited speaker session today. Thank you very much for your active participations during the conference, and we will continue with parallel session this afternoon. Wassalamualaikum warahmatullahi wabarakatuh. All right, ladies and gentlemen, I would like to inform to all the presenters that parallel session will be started at 15 Makassar time. Jadi jam 3:15 akan dimulai sesi paralel sesuai dengan jadwal yang ada di Book of Abstract. Jadi silahkan dicek kembali Book of Abstract-nya, jadwal dan ruangan masing-masing sudah disediakan. Kalau misalnya ada pertanyaan, boleh bertanya melalui grup peserta atau melalui panitia yang ada di depan. Yang pertama, ada program juga. Oh ya. All right, thank you very much. So I will close this session and I will give to the moderator for the parallel session. Wassalamualaikum warahmatullahi wabarakatuh. Tambahan informasi untuk peserta non-pemakalah. Jadi peserta non-pemakalah bebas mau mengikuti sesi paralel di mana saja. Boleh di sini atau boleh ke lantai 7. Tapi kalau untuk pemakalah, mohon untuk mengikuti ruangan sesuai yang disediakan di Book of Abstract. Terima kasih.